Hi, everybody. I'm John Mulcahy. I'm the relatively new president of Carnegie Science. Been on the job about 100 days. It's been a really interesting 100 days, let's just say. But I've been at Carnegie for 30 years. And so I had the chance to know Joe myself and have many conversations with him. So it's so great to see so many of you here today to celebrate an extraordinary individual. We've unfortunately had too many of these at Carnegie recently, but it shows a sign of our success when we have our scientists do so well, that so many people show up. And Joe's career spanned 5 decades, and about 40 of those years were spent at Carnegie. And in many ways, he's kind of almost the perfect Carnegie scientist. The thing about being a Carnegie scientist is you get to do a lot of the work yourselves. When you work at a university or other places, often you hand a lot of your work off to your postdocs and your students. You're busy with many other things. Carnegie is a place where we get to kind of keep our hands on the action. And that's something Joe really, really loved. And I think it was demonstrated through all of his work. Carnegie, of course, is also a place that has had truly tremendous scientists. I'm an astronomer. We've had famous astronomers like Edwin Hubble, Vera Rubin, two of the most famous astronomers of the last 100 years. We've had famous seismologists like Charles Richter, you'll know from the Richter scale, and of course, other famous biologists like Barbara McClintock. And I think it's fair to say that Joe belongs in that class. Absolutely. You can clap for that. Yeah. Because he really is the father of modern cell biology. And I have to say that as an astronomer, I know very little about cell biology, but it was fun to talk to him, and I always enjoyed those conversations. One of the other things I think I want to say is that I think Joe was also pretty revolutionary in mentoring and bringing up the next generation. And particularly, he was one of the earliest people to actually help bring women into science, which is something that also I think is, just clap for that. Because he was doing this in the 60s and 70s when it wasn't really popular or very well known. And of course, even today, the last month, I think a lot of us are seeing challenges with that in that space, which I'm sure he would not appreciate. So I think he was just revolutionary in both of these ways. So in many ways, to me, just really kind of the perfect Carnegie scientist. So I'm going to wrap up there and just say thank you all for coming. We really appreciate. We're going to hear a lot of wonderful things today. And with that, I'm going to bring up Diane, his wife.
So before I begin, I wanted to thank everyone for coming and also to thank the planning team, which was really instrumental in getting all of this together. And that would be Yishen and Alan and Anna and Navid and Natasha, David, Ed, and Jeremy. So a good hand for the getting us all here on time together. Okay, someday you'll be a scientist. Now, that's something that Joe Gall wrote in the year 1942. Someday you'll be a scientist. And so that's going to be the focus of my presentation today. And so I'm going to focus a little bit on where did it all begin. I think you've already seen the pictures of Joe and the microscopes from the time he was a youth in his bedroom to his graduate work to Minnesota, in Minnesota and Yale, and then Carnegie. And I think it's actually more like 7 decades. But I think family was also, you would recognize, as very important to him because he always credited his mother, Elsie, with, with his mother, Elsie, as being one of the people who really supported him, guided him, and inspired him in all of his endeavors in the field, like butterflies and discovering nature. But I think more of his family is also important to see here. You can see his father, who helped get the microscope during wartime, and also a microtome that we'll talk about a little bit later. And also his older brother, seen here, who is the one who sent Joe the book, or handed the book of E.B. Wilson's The Cell, and told him, read this, it's important. And then his mother and Joe shown here again. But beyond that, also the rest of his family was important to him too. And so here are pictures of Minnesota with Dodie and the two children, Larry and Barb. And then when we went, this was our 30th wedding anniversary, where Barb is now with Stu and two girls, Lily and Shelby. Lily and Shelby. Larry has married Nancy and has a granddaughter, Jennifer. And they all were a huge part of our lives for the really 42 years that we were married, and more than their own lives, the children. But I think our family was much bigger, really. It was all of you. And I think this was seen both in the laboratory as well as in homes where we had meetings in our home, and also when we went out to eat. So I think that you're a big part of the family that made Joe himself. And also there were other people, so that some that you might not recognize, like Joe helped with the BioEyes teaching at Carnegie. And also we would often bring friends into the lab to have an exploratory time to see what scientists actually do. Okay, so we've been through microscopes and family. Well, how about dogs? Joe always loved dogs. And in fact, this was one of his favorite things he would tell people is, I've never not had a dog. And that was, in fact, basically true. So here are our dogs over the years, which I think many of you may also recognize, from Tanner, Bridget, Bandit, Cricket, Bentley, Jiminy, and Tyson. But where did all this come from? Where did his love of dogs start? So I think it started probably with his dog Sample, and in 1934, about there, which I think is really interesting just to watch his position. I mean, look at him hold that dog and see how he holds the other dogs. Always almost the same. Okay, so I'm going to go back to where did it all begin? Because you've got three things, but there's a lot more that was Joe. So I'm going to posit that his Christmas card that he wrote to his mother at the age of 14 in 1942, he said, I predict Merry Christmas and a Happy New Year. And modestly in the bottom said Joe. But I think that if you look over on the left-hand side, or your left-hand side, let's focus a little bit more on what he said in this card. This was a card that actually moved, and you could turn the wheel, and there would be 12 predictions, one for each month of the year. And he was writing this for his mother as predictions, but I think he was really doing projections for himself. Water. So I'd like to walk through a few of those. Okay. You will catch bugs all your life. Now, was this for his mother or was this for Joe? And in fact, Joe did that. So here's Joe with an insect in 1976. We go on to being rather bold in Costa Rica and surprising everyone that he would be so bold as to pick these up himself. Polyphemus moths that were mating on our front porch. Those were always a favorite. And then we raised the eggs and released them. Or Joe with the Sphinx moth. This was right outside of the front of Carnegie just a couple of years ago when Joe picked up the Dobson fly, which not a lot of people would feel comfortable picking up, but he knew that it wasn't going to hurt him. And this was Joe, the praying mantis in our backyard that jumped on the back of his head. He loved praying mantises. And then Joe with the monarch butterfly. Each year we would try raising the eggs from our milkweed and then raising them and releasing them. Okay, I think there were some more predictions in that card that he sent. You will be able to retire soon and build yourself a fish pond. Now, he was only 14, so he was writing this to his mother, but I think certainly he was never able to retire soon. but he did build, or he was always near a fish pond. So here he is at his pond. This was the first pond that he had. It was a decorative pond in the garden when he was about 5 years of age. And they lived in Arlington, Virginia there. And this sold him on the house that there was a pond in the yard. Now, I also was looking through the books. I brought some of his books to give away today, but I found a really small one here, and I looked at it, and there's nothing on the spine that you can read, and it's rather dog-eared. I thought, oh, maybe it's a prayer book, or maybe it's a small Bible. So I looked a little bit more, and in fact, it's Field Book of Ponds and Streams. So it was Joe's Bible in a way, and it was by Anne Haven Morgan, PhD, professor of zoology at Mount Holyoke. And it is very well worn. In fact, it opens to a page that has both a dragonfly and a Dytiscus beetle on it, which were two of his favorite. And I would say also that probably to know that there's a professor of biology who was a female writing a book that he so admired probably also had an influence on him over the years. But so from there, this was when he was in the 5th grade, and he was asked to write a book about animals in school. So here he says, "The pond where I raised my frogs," page 52 to 53. "I also raised goldfish and a few water plants in it." And then he took the pictures of the frog here and says, "One of my frogs on a table. This is one of the frogs I raised myself." And then the family in 1942 moved to Upperville, Virginia. So you can see this is a lovely pond that was a little bit bigger and probably had a little bit more of what he was looking for. And this is where they swam. So Joe's on your right. But around when he went to Yale, he found another pond, and this is where he took students to go and explore up in Massachusetts. And this looks a little bit more like Joe rather than in the swimsuit, in his hip boots and trying to find things in the pond. And then here in Baltimore, we found the Park School, which we took people to most every year over about 40 years. And he loved this pond. And here we have that we had the wood frogs that were mating. We would go to listen and see them every year, and then collect eggs, and then the tadpoles here. Now, these actually, for those of you who know, might say, well, those don't look like wood frog eggs. And they're in fact bullfrog eggs, and they came from Barb's pond, but she's going to talk about the pond that he had here near Baltimore. But each year we'd take people there, and Joe would pull out his hand lens and look to see what stage of development the embryos were in, and always bring them back to the lab because he thought that a lot of people didn't actually see the process of development. And everyone was always delighted. And here we have a couple more from our lab that were out with us. And here's Melissa carrying the eggs back. Okay, so we've gone through ponds, bugs, but here's one for those Latin scholars. When he was 14, he writes, "Labor omnia vincit." Remember this, which means hard work conquers all. So I think that you'd attest to the fact that Joe really embodied that. And these were drawings that he did when he was about 1944, so when he was about 15 or 16, which he found how much hard work it was, so that this one, it was enough hard work that he skipped the body. And this dragonfly only has one pair of wings, not two pairs. And he did other detailed drawings as well. But I think his first research proposal actually was in 1943. He actually wrote from Woodbury Forest, where he was in high school. He wrote a 4-page letter to his father, and he said, "After working with my microscope and preparing slides for over a year, it has become evident that I must have access to a microtome if my studies are to proceed past the beginning stage. 90% of all microscopic work is done in thin sections of tissue." Very convincing. And then he said, if I'm going to be a biologist of any worth, I want to get all of my training as early as possible. Now, what father would not give in to that? So he got the microtome. And by that summer, this was what he was doing. He was able to do serial sections that are quite perfect. In fact, this was a toad tadpole in sagittal section that went all the way onto 2 slides, all the way through the body, or the ovary of a lily, which is here. Okay, so Joe graduated from Yale High School and then Yale at 48 and went on to graduate school at Yale and worked hard. Okay, but other things happen. Other aspects of life is play hard. So in this picture that Zara sent me is her 88-year-old boss, Joe, showing no mercy on her playing ping pong at the Carnegie. So where did that come from? So here's a picture of Joe practicing when he was young to be the perfect ping pong player. And here at baseball or softball. And this is at age 95. We were playing pool at the senior center just about a year ago. And what about Joe's love of books? So this is Joe examining books in Mendel's library in Brno in 1995. And I think you've also seen the other pictures of Joe around that time. But I think that it started very early. So this is a book of, with Joe in about 1932, all the way up until to his mid-90s when Joe was showing Staluti's book, The Study of Bees, to colleagues at Carnegie. And he collected over 500 volumes of rare books and kept them in our library at home, as in this shown in this picture. And they're donated to the American Philosophical Society. And you'll hear more about that from David Gary a little bit later in the program. So here's the final one. Remember, I started with someday you'll be a scientist. So I'm going to let Joe speak for himself.
So a lot of it, I think, in retrospect, I think a lot of the value of what I did as a child was that basically everything that I learned from the book and by doing myself. In other words, I didn't have lectures or instruction. People didn't tell me what I should know or what I should find out. I simply used the books and my free time, which I had a lot of, to become a scientist on my own, which I've always felt was very valuable because it gave me the sense that if I wanted to study anything, I could just go and do it myself. I didn't have to have somebody instruct me on how to do it, or I didn't have to become a professional in order to do something that was interesting. So I think that certainly gave me a lot of self-confidence and independence as an investigator, because I was doing independent research by the time I was 8 or 10 years old.
So again, I'd like to thank you all for coming. And Joe was always wonderful. So thank you for being part of his journey. Thank you. I'm going to introduce Barbara Idell, Joe's daughter.
I'm a little short, so. For those who don't know me, I'm Barbara Idell, Joe's daughter. And I don't regularly prepare and present lectures, so my memories to share are more kind of my spontaneous thoughts when I sat down to attend this event. I only have a few pictures to share while I talk, but they all say so much. This is a recent picture of my brother Larry on the left, myself and my dad. As Diane mentioned, family was very important to him, and I know that he considered everybody in this room family as well as his immediate family. So thank you so much for everybody who made the trip. I know some people made very long ones, and for being here today. Everyone in this room was incredibly important to my father, and I know he cared deeply about all of you. So not just about your scientific work and careers, but about really everything important in your lives. And I know this because I heard him relay stories and talk about celebrations as well as challenges that everybody who spent time in his lab faced. So it wasn't just about the science. So this is a pretty amazing gathering for remembrance for all the groundbreaking scientific discoveries in molecular genetics, how they open new avenues of experimentation for scientists around the world. There are many brilliant minds working on deciphering and understanding the building blocks of life. But there were qualities about my father that set him apart, that made him different and made it possible for him to influence more budding scientists. He never once thought to keep discoveries and techniques to himself, because why would you? The focus was always on advancing knowledge in a field that he loved. He wanted to share what he discovered. And I know that he always gave credit to whom it was due. So no wonder you all flocked to his lab. So many of you heard about my father's qualities if you attended the funeral, but they were worth repeating as they really defined for me who he was. Whenever someone points out some of these qualities in me, I feel incredibly proud, because when others see some of these attributes in me, I know they were modeled for me by my father. So without further ado, nothing I'm going to say here will come to as any surprise, but I do have some anecdotes and memories of my childhood that may flush them out a little bit. So he was incredibly smart. He was very logical. He was very thoughtful and kind. He was generous with his time, and that included both in the lab and at home. He was really patient. I rarely saw him mad or frustrated, rarely. But a memorable time was when I was in high school, and my high school guidance counselor told me not to bother pursuing veterinary medicine because not many girls get admitted to vet school. So I think that's probably about enough said. I had to physically keep him from going in there and embarrassing me at 16 or 17 and having a stern talking to. So as you can see, it all ended well. For those who don't know, I'm a veterinarian now. So he endured many, many hours and tears helping with me with my Latin and math homework, which I'm sure he still could have done at 95. Sticking with me until that eureka moment of understanding. Never once frustrated by how long it took. And I'm serious when I say he could still help me with Latin homework. So I know that for sure. He was very, very kind. Harsh words were never really in his vocabulary, and I strive to be that way in my own life. Everybody here knows he was humble, and that he was just doing, when you asked him what he loved, he said he's just doing what he loved. When asked about his accomplishments, he said, I'm just doing what I love. I've never worked a day in my life. I don't know if anybody's heard that, but I heard it again and again. I've never worked a day in my life. So how many people can say that? He was very, very curious. So we've talked about nature a little bit, and I'll just flip the slide now to our pond. And everything in nature was a wonder and something to be investigated and understood. We spent many, many hours walking around our farm pond on our property, collecting specimens or just observing the beauty of a dragonfly. When my husband and I went looking for a house, we love nature too. We found this property with 14 acres and a farm pond, and we both looked at each other and said, well, if it's got 4 walls that are standing, some water and electricity, we're going to take it because this is pretty amazing. So we have this lovely 1-acre farm pond. And then the second thing I thought was, you know, we're gonna live here, and it's wonderful. The second thing I thought is how much my father would like it, because ponds were so important. So we spent many hours. My children, my 2 daughters, spent many hours. In that photo, you can see there's my husband Stu, myself, Dad, our 2 dogs, and my daughter Lily walking around the pond with some beautiful reflections. So the curiosity was always there. And in terms of being curious, he's the only person I ever knew who used a smartphone the best way possible. So it was really just a handheld Encyclopedia Britannica. So he was always using it to, you know, when someone pulls out their smartphone, we tend to get annoyed and wonder what they're looking at text, whatever. But when he pulled it out, he was looking up bird calls on Merlin, anybody who has that app, plants, lichen, animals, just checking the scientific names, make sure he could recall it. So I've never known anybody to use a smartphone, I think, as it was truly designed. No Facebook, TikTok, doom scrolling, none of that. So the other thing we talked about he was a mentor, and I know he mentored each and every person in this room. He mentored me in my life. I've have that drive in myself. I now mentor others in my profession. It's really one of my passions, to share my experience with younger veterinarians and help them mold their professional persona. But it's vital for any next generation of any profession to feel the passion of your work from a mentor and share your time and knowledge. So I know he freely gave of that. So I do have some childhood memories that I just kind of free-formed, and I hope you'll bear with me. There's some that had just stick with me and just come up again and again. So Dad was always working on a manuscript of some sort. And what used to lull me to sleep when I was a kid was the typewriter clacking rhythmically every single night as he wrote his manuscripts, typed things, experiments up, et cetera, because my bedroom was above the study and it used to lull me to sleep. So whenever I hear a typewriter, that's what I think of, which you can't hear anybody on a keyboard right now, but how excited I always was to share my achievements, knowing that he would celebrate with me. Dancing in our living room at midnight on January 1st every year. Writing and solving clues for presents at Christmas time. It was incredibly hard to stump him, but also vice versa, because we operated on similar wavelengths when it came to clues. So anything was on the table. You could write a limerick, you could write a poem, you could write clues in foreign languages, you could write puns, you could intersperse some Latin. I've passed this down to my children, one of whom is here, Lily, who won't accept a gift without a decent clue to work on. And several of you in this room have been subjected and possibly infected with this tradition. So I hope it carries on. It came about to slow down my brother and I opening Christmas presents on Christmas morning, and it worked. So I've often wondered why our handwriting was almost identical. I'm left-handed and he was right-handed, but there it was. I remember the simple pleasures, like knowing how much he liked shortbread and maple syrup and giving him those as a regular gift at Christmas time. Finding the perfect frog every year for a gift for birthdays and Christmas. And believe me, it got harder and harder. And everywhere I go, I still pick up frogs and say, wouldn't Dad like this one? And I know there's going to be some frogs available to look at afterwards. Again, saying to me, time unclear, don't know when I first heard it, that he was the luckiest man because he felt he'd never worked a day in his life. He got to tinker in his lab all day long. As we just heard, Carnegie is the place to do that. And I know he landed at Carnegie because of the rigors of academia and the pulls of the business side of it at Yale were weighing down on him. He wasn't getting to tinker in his lab. I remember how excited he was when Zeiss custom ground microscope lenses for him because of his vision issues. And the day I realized the enormous impact on the global scientific impact of the discovery of lamp brush chromosomes and in situ hybridization. I was sitting in freshman biology class with a slide of a lamp brush chromosome on the screen in large letters crediting Dr. Joseph Gall. my very new friend as a freshman, and the next week jokingly asking me if there's any relationship. That person's still my friend. Watching a total solar eclipse with the newly acquired Questar telescope in North Carolina in the 70s. I had to look up when it was. And a high school science project where we made long exposure star trails, developing the film and printing it together. We now have that Questar telescope. Several weeks ago, my husband and I set it up on our back porch and observed the moon. It's still in good order, will continue to be treasured, and anybody's welcome to come out and look through it as well. He instilled in me a love of words and puzzles, which serves me really well when etymology or word categories come up in Jeopardy. Or doing the Sunday New York Times crossword puzzle, which we did a lot together. So I remember passing around appetizers in my home in Connecticut growing up to many of you, and eavesdropping on the topics of conversation. That was my job, and I loved it. So again, with the pond, that started before we had a pond. Sunday was for walks in the woods in Connecticut when I was growing up. There was requisite binoculars, nets, bags for specimens. We turned over every log in case there was a salamander hiding underneath. And also don't forget the pen and the pencil and the 3x5 cards in the shirt pocket. The ones that used to fall out when he sneezed, because it was always 10 times or more. So my husband actually told me the other night when we were talking about this presentation that he started carrying 3x5 cards in his pocket and a pen and a pencil too, because my father did, and it was such a good idea. So those Sunday walks turned into nature walks around our pond. Some of my favorite memories are watching my daughters catch butterflies around the pond with him, collect pond muck to look under the microscope. Much pond muck was brought into grade schools and middle school and high school when it was a science project due. And when asked what I did over the weekend with my colleagues or friends, well, I collected frog eggs and dragonfly larvae with my dad, just so he could study their chromosomes. So try that with your non-science friends. Another fond memory is watching them stroll along the Delaware beach when my girls were young, teaching them all about horseshoe crabs, because we made a special trip there when the horseshoe crabs were there for mating. And they especially liked the part about the blue blood. Other things that I couldn't replace my childhood is knowing what an axolotl was at a very early age and how to spell it. Traveling through England and Scotland during a sabbatical trips when I was in 4th and 7th grade. And we took many, many trips around the countries and seeing the wonder of Stonehenge, castles, bottle dungeons, Hadrian's Wall, the Bayeux Tapestry, Dover Cliffs, with my father, with just everything with a sense of wonder. And, I realized that not every person gets to do that. So attending the Lasker Award ceremony, that's another big milestone as well. And I know many of you are there as well, being so incredibly proud to be part of that family. Every time I take care of my horses, I remember him saying, they're too big. They have a mind of their own. No one should ever get on their backs. Yet somehow, eventually, buying my first horse. I know Sue Jerby probably had an influence on that, so I thank you for that. Anybody who knows me knows how passionate I am about horses as well as dogs and cats. Hearing him say, if you're going to do something, do it right and have the right tools. So, and then of course, love of books, we talked about it as well. And then fairly recently, traveling to the Galapagos with my dad, Diane and Stu, and reliving Darwin's astounding discoveries through his lens. That was pretty special. So he's left an incredible legacy. I don't want to take up any more time from this, but just a couple things that have been passed on to me is pursue your passion single-mindedly. Don't let anyone discourage or dissuade you. Share your achievement and accomplishments freely with others. Take time to teach and mentor whenever you can. Don't be judgmental. Always foster your sense of wonder and curiosity of the world around you. Every day I see something in nature, like our cattails emerging from our pond after winter or some planetary alignment, and I think Dad would appreciate it. I'm often captivated by new facts in the same way he was. The more intricate and detailed, the better. So, and every week we find something that my dad would love, whether it's an unusual insect or plant. I tell Stu, and we both agree that my dad could explain the facts to us about almost everything. The rest of us must look it up. Yeah. So I'm also incredibly blessed that I had my father in my life for so long and that we were close. He was able to share in all the milestones in my life, as well as my children's lives. I have two grown daughters who really got to know him. It adds depth and perception to your sense of family. I knew my grandparents well, and I know what that feels like. It influences how you became who you are in this world. So thank you again to everybody who came today in person or virtually. I'm looking forward to hearing everybody's sharing about favorite memories, vignettes, quotes, achievements, and just to celebrate my father's influence in the global scientific community. Everybody here is his extended family and his legacy. And this is just a beautiful sunset over our pond, which we get to look at every day, and that's Turbo. Yeah. Yeah. So.
Well, welcome. I'm Alan Spradling. I'll be introducing the rest of the speakers after those two wonderful presentations. I knew Joe Gall for more than 50 years. Yet even when I first met him in 1974, he was already my scientific role model and hero. He occupies a similar position in the minds, I think, of many of those of us who are here and scientists around the world. To celebrate why Joe and his science are so special, we'll hear from faculty colleagues and lab members from each of the three institutions that he was at during parts of his career. He joined the faculty at the University of Minnesota at the age of 34 or 24, even before his first paper was published. Try that today. 10 years later, he moved to Yale as a co-founder and exponent of cell biology, an old discipline that he and a handful of others had renamed and were upgrading. They had recently founded this American Society for Cell Biology, stimulated initially by the advent of electron microscopy. After 20 years at Yale, Joe moved to Baltimore, where he spent the majority of his career here at the embryology department. For more than 40 years, he continued working at the bench and thrilling biologists with his new discoveries and insights. In addition to today's formal presentation, we look forward to your own thoughts during the open mic session. We want to share your stories with Joe's family and friends and preserve them with his papers and book collection at the American Philosophical Society. So for many of us, myself included, Joe Gall epitomized the scientist that we wanted to be. The characteristics of such an investigator are widely acknowledged. Someone who discovers unanticipated aspects of the biological world and whose findings and creative ideas clarify and extend understanding. Joe's science did that and also had a further element. He loved to present great science in a manner that also highlighted the sheer physical beauty that suffuses biological materials. When I was a graduate student, my initial studies of the genes and cell that a cell expresses contained some truth, not much, and very little beauty. Oops. That tendency to go in the upper button is strong. So here's a sucrose gradient, and it's showing you the distribution of sizes of all the mRNAs that one cell makes. And You can't tell what mRNAs they are. You can't really tell how many different ones there are. And you can't tell what any of them do. And I tried to get excited about that, but it was a little bit hard. I actually never heard too many people say that sucrose gradients or bands on a gel were beautiful. However, coming into the next phase of my graduate career in Mary Lou Pardue's laboratory, I came into Joe's sphere of influence. And I was introduced to the giant polytene chromosomes of Drosophila, shown here. These were the very structures Joe's graduate mentor, Don Poulsen, had tried to use him to lure him to study in his lab. Joe had declined. He was already very comfortable making his own scientific decisions, and he thought polytene chromosome work had plateaued. So instead, he decided to work on the much lesser known giant lampbrush chromosomes, and we all know where that went. On polythenes, however, for me, I could visualize thousands of bands and learn that each one had intricate structure and distinctive aspects. With practice, this made it possible, using just a few bands, to identify the exact chromosome location from which they derived. This reproducible complexity made these chromosomes seem incredibly beautiful and mysterious. And one had the sense that they held countless undiscovered truths about the genome and about how it works. Looking at them made you feel, as Barbara McClintock once remarked, that you could climb down into a field of chromosomes and marvel at them as you walked by. Joe and Mary Lou's discovery of in situ hybridization made the beauty of polytene chromosomes reveal more of their hidden truths. It firmly moved the field off the plateau that Joe had worried about at Yale. With in situ hybridization, you could map where every piece of genomic DNA was located relative to any other piece. Amazingly, it also allowed you to start learning about gene regulation. You could label cellular RNA and hybridize those thousands of mRNAs to a polytene chromosome. And of course, only the most abundant and well-labeled ones would make a band on the chromosome that you could identify. But you could see the number of those bands and where they were located. And then if you did this, say, under two different conditions, they might not be exactly the same. Here's an example from those days in Mary Lou's lab, where here's some polytene chromosomes, and I know it's a little hard to see, but if you look down here, there's some bands are very heavily labeled, and then others are lightly labeled, but they're reproducible. And with effort, you could map over 50 genes that were present, and this is if you took the cells first and heat shocked them for a few minutes before adding the label and preparing and doing the hybridization, compared to if you just had a control where you hadn't heat shocked. And not only that, a lot of the bands that were in the control were no longer present after the heat shock. So you really could see that the cell was responding quickly and changing its gene expression. But amazingly, that wasn't really known, that cells were so quick to change their behavior in RNA production. So combining classical and molecular biology, as is epitomized by this experiment, was a signature characteristic of Joe Gall's science. For example, Carnegie Genetics staff member Calvin Bridges, many years ago, identified more classical Drosophila genes just by their mutant phenotypes than anyone else. And this information was mapped to appropriate but incompletely accurate locations and published in many publications from the Carnegie Institution. This work actually represents the first genome project, and Bridges was acknowledged as the greatest master of polytene chromosomes. Dissatisfied actually with the photographic guide that most people were using, Joe once made his own photographic guide to the Drosophila palatine chromosomes. I still have a copy in my lab, and I would occasionally use those instead of the more traditional one. The Drosophila community embraced in situ hybridization and the palatine map and used it to zero in on all the genes in the genome. Combined with insertion mutations, it became really possible to start to identify the nature of these hundreds of bridges, mostly, and other people's identified genes. And this final slide shows you a little bit about how this process worked. So here you see that if you have an insertion mutation and you do in situ hybridization with a homologous sequence only in that element, you get a very nice signal on one precise band. This whole region is just 1% of the palatine chromosomes, and you can see a tremendous resolution. This particular one, 3999, which you see here, this is just a tiny page from a collection of all these locations. And here you see the ones where the gene has been identified. And here you see that this insert is at a gene that Bridges called stripe when he first discovered it. Here's a picture of it up here. I know you probably can't see this, but there's a very faint grayish stripe on its back, which shouldn't be there. So that's a mutant phenotype. And this was in 1923. And sure enough, in Carnegie publication number 327, which a picture of which is shown here, there was a description of this discovery and information about the mutation, along with hundreds of others. But these would have just languished if in situ hybridization and molecular technology hadn't come along and made it possible to start to get to a deeper level. And so stripe, you know, from this, possibly from this insertion, I didn't really look back, was found to encode a transcription factor, and it's said to have something to do with tendons. And I don't know if that affects the flight muscles or why it leads to this stripe. And that's characteristic of many of the genes. Even as you make more progress and learn much about them, there's still a lot left to learn about what's going on. So how did Joe partner with so many diverse people, carrying out impactful and beautiful science continuously throughout a 70-year career? How did he make clarity and creativity seem so natural and straightforward? Joe managed to achieve complete independence from any goal other than a search for truth, as judged by his own exacting standards. Certainly, there must have been multiple factors, but I believe one of the biggest was his early and continuous intellectual independence. He might accurately say, I've been managing my own research since I was 12, and I guess I heard earlier that maybe I should have said 8 or 10. Most of his mentors, including some of the great early innovators of cell biology, he knew only through his writings. The critical role they played in his training may be one reason why he became so interested in acquiring their books and learning about how they made their discoveries. And the interview that Diane just played for you seems to really confirm that that was also his view of things. Joe readily admitted that most scientists don't know history of their subject and don't need history to do good work. You don't need it. But Joe made a convincing case that there are at least some times when it can be a tremendous advantage. In closing, as we celebrate Joe's accomplishments this afternoon, I hope you'll be inspired by the example to take another step on your own path to great science. In doing so, you will honor Joe in the same way that he revered the truth seekers whose lives he treasured and who guided his own intellectual journey so unerringly. Thank you. And I'd like to introduce our next speaker, Joan Stites. Joan grew up in Minnesota and was in Joe's lab for a period of time when he was there. she went on to be one of the most distinguished studiers of RNA in the world, discovering very fundamental things about how messages and ribosomes get together, how R and P are combinations of small RNA and proteins that guide RNA processing, splicing, and the modifications of bases. And she's going to tell you, her experiences with Joe in those days. Joan?
Wow. I'm indeed greatly, greatly honored to be here. Joe was fantastic. And I feel embarrassed in a certain sense that what I'm going to be telling you about is mostly about, if I can get, oh, wait, the next slide. Wait, wrong way. No, that's not me. There we go. Okay. Is not really mostly about Joe, but it's mostly about me. And it's about how his influence impacted at least one of the people with whom he came in contact. And I know there were many, many, many of you that have similar stories that are equally important for your lives as my running into Joe serendipitously was for my life at a very early time. So let me take you back. This says how serendipity and Joe Gall fashioned my life. The serendipity starts in everybody's life, I think, very, very early on. And for me, I was fortunate in having been born to two parents who both believed that women should not just have babies, but also have careers. And as a result, my interest in science as a child was very much encouraged by my father, and that turned out to be incredibly important. When it came time for deciding where I might go to college, I actually could have gotten into and did get into many different places, but it was decided, and again, because of this thing I've already told you about, that I would go to Antioch College, which is a liberal arts college in Ohio that some of you may know is unusual in that it has a work-study program. And what that means is that instead of students spending 4 years on campus taking courses, every other, as it turned out, quarter, students would leave campus and leave their books and go out into some place in the world. It could be in the US, it could have been anywhere in the world, and do a job that in most cases was relevant to what they might be doing, what they thought their interests might be. And so it was through this sort of program that I first got introduced to molecular biology before I even ended up in Joe Gall's lab for the summer of 1963, which was the summer after I graduated from Antioch. Let's see, no. So I think this isn't, okay, I'm not doing well on this. There we go, okay. So up there at the top is formative experiences, Alex Rich's lab at MIT. So I was fortunate to end up, due to Antioch's work-study program, in the lab of Alex Rich, who was one of the 30 or so people really sort of doing, quote, molecular biology at that stage. This was the early 1960s. And the way that happened, man, I just want to tell this story, and I'm going to tell a couple little stories like this, was that there had been a student a couple of years ahead of me who was a psychology major who had ended up working at the Yale Child Study Center for one of her co-op jobs. And there she had met a young biophysicist who was working in a newly built building on Science Hill called Gibbs Lab. And they had fallen in love. So when he moved to Alex Rich's lab to further his postdoc, she wanted to be in Boston and had figured out that it would be good to have a job in his lab. So that became a lab job then that was passed on from one Antioch student to another. And I should also just mention there, for those of you who happen to know Roy Parker as one of the luminaries in the RNA field, his father, Bob Parker, Robert Parker, was the head of this Antioch co-op program that assigned students to various labs as they moved on through their careers. I arrived in Alex Rich's lab in January of 1961. So this was 8 years after the double-stranded structure of DNA had been published. However, it had not yet made its way into textbooks and not even really into discussion in courses. And I remember being totally enthralled at the point that I learned about this, because for the first time it offered a possible molecular explanation for how heredity might be passed on through generations through a molecule that self-replicated. And I was completely taken over by this. In Alex Rich's lab, oops, why can't I do this the right way? There we go. Okay. I ended up working with a graduate student, shown down here, John Warner, who many of you will know as a person who spent the majority of his career at Albert Einstein and was very influential in discovering how ribosomes are made, particularly in yeast cells. And we were doing an experiment, which was sort of an experiment that you might do in those days, because new nucleic acid molecules were being found associated with various cellular organelles. And this was the experiment I was supposed to do, was to take ribosomes. And it was known that if you heated up a ribosome solution, that the hyperchromicity would go up, it would absorb more light as the RNA denatured. And it was known for DNA that if you then reduce the temperature, the absorbance would go back down, meaning that the DNA had renatured. So if DNA can do this, why can't ribosomes renature? And I spent a very, very long time standing fruitlessly in front of the DU spectrophotometer, heating up solutions of ribosomes in various buffers and cooling them back down, and of course never got them to reform, because we now know ribosome preparations are contaminated with RNase, and they just chew up the RNA the minute it's released, and there was no hope for the experiment. Nonetheless, I was still very, very interested in the field. The next year I was to go to Europe and actually spent some time there working in the lab of Alfred Geer in the Institute for Virology in Tübingen, which was a wonderful experience. And during the year, so that was my sort of last year of courses. And during that year, after debating a lot between medical school and graduate school, I decided that I better apply to medical school. And this was because I looked around and I didn't see any women professors or heads of lab. I did see a few women physicians, and therefore I decided that this was what I should end up doing. So when I graduated in June of 1953, I wanted to be home with my parents for a bit of time. I knew that there were people at the University of Minnesota, including Joe Gall, who were doing interesting things. And I went around and talked to 5 or 6 different professors. Would they take me into their lab for the summer with this background that I had from Alex Rich's lab and in Europe, et cetera. And the only person that said yes was Joe Gall. So I entered Joe Gall's lab right after graduating from college. And the project that Joe had me working on, oh, actually, no, I'm not going to get there yet. Okay. Okay. Anyhow, let me tell you, the project that he had me working on was, again, one of these, is there nucleic acid in various cellular organelles, was isolating basal bodies from Tetrahymenopiroformis, and then analyzing them for both their DNA and RNA content to see if there was any evidence of nucleic acid being there. I'm not sure I would believe the results that I now, that emerge from that nowadays, given what I now know. But in any case, during the summer was also a time when one day, oh, come on. No, that's not mine. Oh, wait, yes, it is. Okay, sorry. It looks very funny from, can you see the actual ellipse shapes? Yeah. One day Joe grabbed his camera and he said, okay, we got to go outside and take a picture of you. And this was a partial eclipse of the sun. And that's the picture he took of me in August of 1963 outside of his lab. By that time, even though Joe's lab had been very small, there was one other undergraduate student, there was one postdoc whose names I cannot remember. There was also an EM technician whose name I do know. And Joe mostly spent his time packing his boxes to move to Yale. Nonetheless, I got turned on by this project I'd been assigned to do all by myself, typical Joe Gall fashion. And by the middle of August, I decided that I really didn't want to go to medical school. I wanted to go to graduate school. And could Joe help me? And this is just one example of the wonderful things that Joe does for people. But he got together with Jim Watson, who he knew I'd met when I'd been in Alex Ridge's lab at MIT. And somehow they engineered my entry into a graduate program at Harvard, in a brand new one in biochemistry and molecular biology. And I forewent my Harvard Medical School acceptance and that sort of career. As a result of this whole project, I want to say one more thing about Joe. Sorry about this. There we go. A paper did in fact emerge. And you will notice that doesn't say Joe Gall anywhere. As a matter of fact, if you read the entire 4-page paper, Joe Gall's name is not mentioned. But the paper, every word of it was written by Joe Gall. And somehow I let it slip and have felt embarrassed ever since that he did not take any credit for this paper. The technician who did the EM work is thanked in the acknowledgements. And that's as far as Joe would go. So that's just another. So from here, the couple of stories I wanted to tell really have very little to do with Joe, except for the sort of principles that I think I picked up and internalized from him in the very short time that I was in his lab. I went off to Harvard. I did the first year graduate courses. I wanted to decided I wanted to work on something similar to what Joe was working on. And the person who was at Harvard who was big in that field was Keith Porter. And I went along to ask him whether I could possibly be a graduate student in his lab. And Porter took one look at me and said, "Oh, but you're a woman. You'll get married and have kids. What good will a PhD do you then?" And so I went to my second choice thesis advisor, which again was very, very impactful and has done a lot of things, Jim Watson, and asked if I could be in his lab. And that is see if we can do this now. Okay. Going backwards, going forwards. There we go. Okay. Ended up in Jim Watson's lab, which at that time was a sort of hub for molecular biology in the whole city of Boston. And Jim used to have these pictures taken every year on the rhinoceros statues outside of the biolabs. This is me up here with my mouth hanging open. There are some other luminaries in this picture. Here's Jim, here's Wally Gilbert, here's Mario Capecchi, et cetera, et cetera. So you can see the kind of lab that it was. And that was very, very stimulating. I think I've basically finished with stories about me. Let me just say one more. Oh, darn. Oh, Joe. There we go. So by the end of graduate school, I had met my husband, who's a protein crystallographer, so there's no place to go but Cambridge, England, because that was the mecca of protein crystallography. We got there, and Jim had written to his friend Francis saying I wasn't too bad, and they should probably accept me as a postdoc in the lab. And I went to talk to Francis after we arrived, and he said, well, we don't really have a project for you. We don't really have space for you. How about doing something in the library? And I knew that library work was not my forte. So what I did was I went along and talked to all the postdocs in the lab. It was about a dozen. They were all male, most of them from the US, about potential projects, and ended up oh, I can't do this. Ended up, okay, with a project that's already been alluded to that many of you probably know about, which was binding ribosomes to messenger RNA under conditions of the initiation of protein synthesis, and looking at the sequences and seeing what signals besides the genetic code could be observed in those. And then later, many years later, it turned out that work of two Australian workers, John Chan and Lynn Dorgano, came up with the idea of base pairing between the ribosomal RNA and the messenger RNA as a signal for the initiation of protein synthesis. So I'm not going to go through, I hope I can get this right, going backwards again. Okay, I'm not going to go through anything more on here. Just things that we had a lot of fun along the way. Lots of things happened. I wanted to say thanks to all of my colleagues, but especially I wouldn't be here if it hadn't been for Joe Gall and accepting me into his lab that first summer, and then somehow orchestrating my switching from medical school into a graduate program. And I shall be eternally grateful. And I just want to finish with this slide that happened to be in a pile that I found with Mary Lou and Oscar Miller. I don't know when it was taken. I think it was about 1983. I think it was taken at the National Academy of Sciences. Okay, I don't know, but you've heard enough about me. You don't want to hear more. But what I owe to Joe is an entire lifetime in science that I've clearly enjoyed. And thank you all for listening. Oh.
Well, our next speaker is Joel Rosenbaum, distinguished cell biologist who knew Joe at Yale from much of the time he was there. Joel's a famous cell biologist, E.B. Wilson Award winner, best known for the flagellar transport pathway that's both very fundamentally important and also very important in numerous human diseases. So, Joel? I thought I knew everything about Joan Steitz's career, but I learned some new things, and I also learned some new things about Joe Gall's career from Diane Dwyer, which I found very nice to see. I have some stories that I'd like to tell you. There are too many for me to choose from. They all revolve around Joe Gall. I first met Joe when I was a postdoctoral student with Houston Swift at the University of Chicago in the early to mid 60s. And Houston was a good friend of Joe Gall, who was up at the University of Minnesota in the zoology department. He would have Joe down to speak to the cell biology class and also to give a research seminar at Chicago. The graduate students and postdocs in Swift's lab always looked forward to Joe's visits. We had read his work on centrioles, and we had read his early work on chromosome structure, and it was always an exciting time centered around his visits. Gall would speak to all of us individually, graduate students and postdocs, when he came down to lecture. And so it was a good time. And I thought, after listening to him a number of times, that if I ever had the chance to work in a department or situation that was close to Joe Gall, I would jump at it. I had a successful postdoc, and when it was concluding, had some good offers. Universities for offers to give seminars, at least, for positions at places like Harvard and at Yale, among others. Harvard at that time had a great accumulation of cell and molecular biologists. So I was pleased when I got an invitation to speak, to give a seminar at Harvard, and Keith Porter and George Wald, the Nobelist, were my hosts. I gave my seminar and met students like Tilney and Richard McIntosh and others. And before I left, having lunch at the Harvard Club with Keith Porter, he asked me, "Why do you want to come to Harvard?" And I said, "Well, Ian Gibbons is here. And since I'm working on cilia, and it's a rather new and exciting field, it would really be great to be working in the same department as Ian." And Porter looked at me and he said, "Hasn't anyone told you?" And I looked at him questioningly, and he said, "Well, Ian won't be here anymore. He didn't get tenure at Harvard, and we would be hiring you to take his position." That, as far as I was concerned, was the kiss of death for Harvard. As far as I was concerned, if they didn't keep this person that I thought was doing really great work in my field, they certainly were not going to keep me. Yale at that time did not have the accumulation of famous and cell biologists and molecular biologists that Harvard did. And the Klein Biology Tower, the new 12-story building at that time, was just going up. A few days after I returned from Harvard to the University of Chicago, I got a call from George Wall offering me the position as an assistant professor in the biology department. In the meantime, I had learned that Joe Gall, the professor who would come down from zoology at Minnesota and whose lectures I really liked, was taking a new professorship at Yale University. Soon after, I got a call from George Wall saying, "You have the position at Harvard as an assistant professor," and I was pleased to say I'm not taking it. And he said, "Why not?" I said, "Well, any place that would not give Ian Gibbons a promotion is probably not the place for me." And also, I learned that someone who I really respected a lot was going to Yale, and that's where I was going. Wald, hearing this, never after acknowledged my existence. Even though we passed many times on the streets of Woods Hole, where I spent many summers. I knew very little, by the way, about Yale, except that they were making a big push to build cell biology, and the appointment of Joe Gall was the beginning of that push. And to follow were many people, some like Joan, who you've just heard sitting here, came to Yale with her husband, Tom Stites, and eventually Pilati and Jameson, and but that arrived on the scene. But at the time that I got there and Joe Gall was just coming, Yale did not have the reputation that Harvard did in cell biology. That was soon to change. So for me, it was a dream come true. A dream as a postdoc that this person, this kid from the St. Lawrence River Valley in northern New York, would be a professor at Yale and working with someone who he had the utmost respect for. At Yale, Gall soon had a lab full of exciting and bright graduate and postdoctoral students. And my lab was just down the hall from his. His students, for the most part, had their own research project, and Joe Gall had his. We all got together for lunch on the 4th floor of the new Klein Biology Tower, and every lunchtime was like going to a scientific meeting. People chirping in with new literature that they had, new findings in the lab, Susan Jerby, who's here, Ginger Zakian, and others, Mary Lou Pardue. And so it was a fun time and an exciting time. Certainly an exciting time in cell and molecular biology. You've all heard a lot about Joe's research accomplishments and also the many national and international awards that he has been given for those research accomplishments. What you have not heard as much about, and I want to say a few words about it right now, was his ability as a teacher. I taught with Joe for well over 15 years the undergraduate cell biology course at Yale. Literally hundreds of students passed through that class. I went to all of his lectures because co-teaching the course, we went to each other's lectures. And it was a learning experience for me as well as for the students. Each new topic he covered in his lectures was first prefaced by a full lecture on the history of what he was going to talk about, whether it was the nucleus, the cell membrane, the mitochondria, whatever it was, you heard what he was going to talk about. Subsequently, you heard the history. And it was a beautiful history because it was illustrated by figures from old books from his own library. I was never quite sure how Joe knew so much, how he had so much information at his fingertips, and how he could organize it so beautifully into a 50 or 55-minute undergraduate lecture. Once I had the temerity to ask him, Joe, every time I hear you give a lecture, I wonder, how did you do this? And all I got in return for an answer was a shrug and a very shy smile. That was Joe. Joe also had, as we all did at those times, undergraduates working in his research lab. One of them was Joan Argetsinger, now Stites, who was later to achieve fame on her own as a Yale professor. She published, as you've just seen, the first page of the publication, published her undergraduate work done in Joe's lab in the Journal of Cell Biology. Very unusual for an undergraduate to publish in such a prestigious journal at that stage of one's career. Another undergraduate in his lab, now at Yale, was Jean Bennett, now from the University of Pennsylvania, Children's Hospital of Philadelphia. She recently received an honorary degree from Yale University. She was the first in her class that admitted women to Yale in 1970. And later, she was the first to successfully use viral vectors to transfer genes into children with progressive blindness due to mutations in the gene RP65, and to elicit sight in these children, a project spanning over 10 years, 12 years of research. The work is fantastic to read, and Yale honored her, as I said, with an honorary degree. That was Joe's undergraduate student, who later went on to fame. I have many, many stories of life on the fourth floor of the Klein Biology Tower with Joe Gall and his students. One, however, has always remained with me because it illustrated so well the enthusiasm Joe had for research in cell biology and the joy, the sheer joy he got out of doing it, and doing it with his own hands. One day he came running down the long corridor between our labs in the Klein Biology Tower, which had office doors and lab doors opening onto it, calling out, "Come see this, come see this, come see this." And heads popped out of the door, and he was calling us to take a look through the microscope at the first successful autoradiographs of in situ hybridization with labeled RNA, which he had done with his student, Mary Lou Pardue, the first results on the oocytes. That was sort of typical of life on the 4th floor of the Klein Biology Tower next to Joe Gall. It is always sad when a longtime friend and colleague who influenced our lives so much passes on. And I find it hard to believe that Joe is not here anymore, because it seemed he was always there for us. But he has left a lot of himself with us. And for that, I am thankful and I am grateful. We owe him a lot. Thank you very much. The pleasure to introduce our next speaker, Susan Jerby, who was a postdoc with Joe at Yale, grad student, sorry. And she has spent much of her career at Brown, where she's a distinguished professor, and working on an organism that is one of the lesser recognized organisms, but Susan has done a tremendous job convincing people why Sierra caprophila is a really useful and exciting system because of its many unusual chromosome behaviors. She and a postdoc, a former postdoc, John Urban, have worked out the DNA sequence. It's become a very tractable system, and she's convinced an increasing number of people that it's worth studying, including myself. And I'd like to introduce her now.
Well, thank you, Alan, for the plug for Sierra and also for your support over the many years of this wonderful organism. All of you know that Joe had a variety of an enormous range of organisms in his lab, and he encouraged me to bring Sierra with me from my undergraduate work to work on that in his lab. So thank you all for coming today from some distance for many of you to celebrate the amazing career of Joe Gall. He was an extraordinary cell biologist, naturalist, and mentor extraordinaire. I was lucky to be one of his first graduate students at Yale, just down the hall from Joel Rosenbaum on the 4th floor at Klein Biology Tower. And so I've known Joe for 60 years. He was my Dr. Vater, which is a term he used, which is German for doctor father. So he was my scientific father, and I think I speak for all of those of us who were his trainees that indeed he was our scientific father for all of us. So the many aspects of Joe have been reviewed in the past half year in several retrospective articles, and I've listed them here, and many of the authors are here in the audience as well. But I would spend hours if I told you about everything in those articles, so you'll have to go read them. But instead, I'll just dwell on a few of the many facets of Joe Gall. So, as you already heard, at age 14, Joe, with his parents and older and younger brother, moved to Ammondale Farm in Upperville, Virginia. And this afforded him the possibility of roaming through the fields and collecting things both in the fields and in the streams. His mother helped him in this by sewing for him, collecting nets. And this became a lifelong passion, as you see here in this slide. Also, oops, you saw from Diane, where Joe later in life is collecting newts. So Joe had an extraordinary number of different organisms in his lab. As a naturalist, he knew their many properties, and so he could always select the best organism to suit the biological question at hand. You've already heard about Joe's lifelong interest in microscopy. And so on his collecting sprees, he wanted to look more closely at the things he was collecting, especially the protozoa from the ponds and so forth. And so even though it was World War II and things were hard to come by, Joe's parents did manage to get for him a Bausch and Lomb microscope. And he set this up in his bedroom in his home. So at age 14, he had a laboratory that he could work in. And subsequently, when he was at Yale for his PhD work, he needed an inverted microscope to study amphibian oocytes, but those weren't readily available. And so he made it himself, together with the help of his uncle, who was a machinist. You can see on the right that Joe continued microscopy at the University of Minnesota. And in addition to phase microscopy, later on in his career, he did confocal microscopy, and much more recently here at the Carnegie, super resolution microscopy with his STED microscope, which he's collaborated with some of you in the audience on helping you for your work. The other thing you've also heard about already is Joe's love of the history of science and his love of books that had the original articles and illustrations. And here you see in Brunn in Czechoslovakia, Joe is examining Mendel's copy of Darwin's Origin of the Species. And as Joe wrote on the back of that photo, this was taken in September 1995. He collected an enormous number of antique books, and over a period of 5 years, every month, the cover of the Molecular Biology of the Cell journal had one of the reproductions from his own personal collection of historical covers. Those 60 covers then became summarized in the book he wrote together with Dick McIntosh, published by ASCB, called Landmark Papers in Cell Biology. So he went as an undergrad to Yale and thought he was going to be a medical doctor, but in his junior year discovered that you could get a PhD and do research. And so he did a pivot and continued on for graduate school at Yale, working with Don Poulsen, a Drosophila geneticist. And although, as you already heard from Alan, that Don tried to convince Joe to work with polytene chromosomes, Joe felt that everything worth knowing had already been found, or else that things that were worth knowing were too difficult to explore. And so instead, Joe became fascinated by reading Waddington's 1939 book, Introduction to Modern Genetics, that there were these gigantic chromosomes in amphibian oocytes called lamprush chromosomes that had been studied since the 1880s. And so he decided that even though he was in a lab doing Drosophila work, he was going to independently do his own project, and that was going to be to study these gigantic chromosomes. And that became the subject of his thesis. And here you see a clip from the iBio film that Joe narrates, and the lamprush chromosome, shown here, is a picture taken by Mark Roth, I believe. So at the University of Minnesota, Joe started there as an instructor and quickly rose up through the ranks to become professor. I should note that he did not do a postdoc. He completed his PhD in 3 years and then immediately went on to become a faculty member at Minnesota. And at Minnesota, he started corresponding with Mick Callan in St. Andrews and also Mick's student, Herbert McGregor, who were also working on Lamprecht chromosomes. And collaboratively, they found out that the Lamprecht chromosome has 2 chromatids with pairs of lateral loops. And moreover, the loops are sites of transcription. So Herbert had done an experiment using DNase that showed that it would break the loops, suggesting that their axis was contained DNA. And Joe decided on his own that he would then follow the kinetics of DNase breakage, because this could then resolve how many strands of DNA had to be broken. And this led to the proof of the unity model, that is that there's 1 DNA double helix per chromatid. And that was an area of hot debate at the time, and Joe solved it again by microscopy. So at the University of Minnesota, they also acquired one of the first electron microscopes. He shared this instrument with Marilyn Farquhar, whom some of you know. And using this new tool, he was able to show the octagonal symmetry of nuclear pores for the first time, and also that the new daughter centriole arises at right angles to the mother centriole. So electron microscopy, as I said, had just been invented recently, and it led to the founding in 1960, while Joe was at the University of Minnesota, of the American Society for Cell Biology. Joe became an early member, not a founding member, but an early member of ASCB. And 7 years later, he became president of ASCB. Subsequently, he received the highest honor of ASCB for scientific achievement, the E.B. Wilson Medal. And that's especially appropriate because as a teenager at 14, as you already heard, his brother gave him a copy of E.B. Wilson's book, The Cell in Development and Heredity, which greatly influenced Joe during his career. The other notable thing that happened at Minnesota, as you already know, is that Joan was in his lab for the summer. So I almost went to Minnesota because I knew as an undergrad at Barnard College that I wanted to work with Joe Gall. I was fascinated by chromosomes already at that time. But then I heard that he was moving to Yale. And so instead I applied to Yale, and I was in his first crop of grad students at Yale. And at Yale, he was appointed as the Ross Granville Harrison Professor of Biology. And he did a pivot at that point in his career. So previously, he had focused primarily on microscopy. And although that was still his love, and he still continued throughout his career to do microscopy, he self-taught himself how to do molecular biology. So this was a newly emerging field, and he taught himself how you would work with DNA and RNA. And so much of the experiments that he did at Yale revolved around DNA. And then when he came to the Carnegie, many of those experiments revolved around RNA. So one of his early experiments at Yale was the isolation of the first eukaryotic gene. And this was before the era of DNA cloning, so this was quite remarkable. And this was independently done at the same time, in the same year, by Don Brown here at the Carnegie, together with Igor Dawid, who was in Don's lab. And so the 2 papers came out in the same year. And this really ushered in the era of molecular genetics. And the first gene to ever be cloned was, again, the ribosomal RNA genes from amphibian oocytes, which is what is shown here in the original paper of their isolation on a cesium chloride gradient, where the blue shows you the ribosomal DNA, which is a higher base G plus C base composition than the bulk DNA shown in red. So this was also the era where molecular hybridization had just been discovered. And if you could do this in a test tube, then it became intriguing to ask whether you could also do this for chromosomes in situ on a slide. And so many people were trying this, at least half a dozen labs were furiously trying to get this technique to work, but Joe had the edge on them. And the edge was he was a biologist at heart, and he knew what model system to use to approach this question. Because if you got an answer when you're trying to develop a method and you don't know what the answer should be, then you could be misled and you might be just looking at background. But he knew from the emerging experiments at the time, including his own, that ribosomal RNA genes were located in nucleoli. And therefore, the perfect system to use to develop the method of in situ hybridization was the amphibian oocyte, which had multiple nucleoli. And at one stage, they aggregate together, as you see here, to form a cap. And so he knew what he was expecting to see as a result. And so he could refine the method so that it would work satisfactorily. So he succeeded in developing the method at its first stage and getting things to work. And you've already heard about Joe running down the hall to tell Joel that this was working. And at that stage, Mary Lou Pardue, my classmate at Yale, was trying some very elegant but difficult experiments for her thesis work, and it wasn't working. And so she did a pivot, and Joe invited her to join him in further developing the method of in situ hybridization. And so the first description of the method is shown here from their PNAS paper. And then the next step was to see, could you not have the hybridization occur at extra chromosomal nucleoli, but on chromosomes? And that's where I was fortunate to come into the scene as well. And so working together with Joe and Mary Lou, the first chromosomes to be used were the giant polygene chromosomes from the fly Sierra, which I had in the lab. But then the question was, could we do other sequences besides ribosomal RNA as a probe? And again, because one needed to have some repetitive nature for the target, it was decided that it would be a good choice to try to locate the simple sequence satellite DNA from mouse cells. And so Mary Lou and Joe succeeded in doing that, as you see here. And they found that the sequence of centromeric heterochromatin was composed of simple sequence satellite DNA. So you've already heard about using a variety of organisms, from amphibia to insects to cells in culture, and then came along Tetrahymena. And of course, Joe was familiar with this organism because it was in the pond water that he had collected and observed even as a teenager. And so Tetrahymena has fascinating biology where the micronucleus then gives rise to the macronucleus, which is the somatic nucleus. And in that process, the chromosomes break apart into many, many pieces. And those pieces go on and amplify and produce many copies of themselves. And so Joe observed that the ribosomal RNA genes in Tetrahymena were indeed extra chromosomal copies. And with his grad student, Kathy Carr, whoops, now what did I do? They discovered, or Kathy discovered, and then sent Joe a cryptic message while he was in England at the time, that indeed it was palindromic, that is, facing together. And shortly thereafter, Liz Blackburn, who had learned how to do DNA sequencing in Cambridge, came to do a postdoc in Joe's lab. And of course, it would be great to try and sequence something from a eukaryotic chromosome. And the perfect material was tetrahymena rDNA. And Liz found that the ends were G4T2, and that indeed is the hallmark of telomeres for many, many organisms or some variant thereof. So after 20 years at Yale, there was a lot of pressure on Joe to take on administrative positions such as department chair or heading the graduate program. And these would take him away from his work, his own work with his own hands in the lab. And so he was despairing about having his time gobbled up with administrative duties. And at that point, Don Brown came to the rescue and said, invited him to come to Carnegie, where he could continue his research for 40 years. And so at Carnegie, he did another pivot and started working more on RNA rather than DNA. And working on nuclear bodies, he suggested to the field that they be named Cajal bodies after Ramon E. Cajal, who had first observed them in 1904, and then subsequently unstable intronic sequences or cis-RNA. And so Joe continued throughout his career to work at the bench, and he never changes. I mean, here he is at age 74, here he is at age 89, and it looks almost identical, and he's still dissecting amphibian oocytes. And so every 10 years, we would have a birthday symposium for Joe. And at his 85th birthday symposium here at the Carnegie, Don Brown gave the after-dinner lecture. And he said, most scientists continue on an upward trajectory until maybe they're in their 40s or 50s. And then it kind of plateaus, and then it starts to go downhill. And then he had a slide that he showed, and he said, but not Joe. Here, the trajectory just keeps going up and up and up and up and up. And this was the 85th birthday celebration for Joe. And so Don concluded, Joe must be a mutant. Well, in recognition of Joe's many accomplishments, he's received many awards that are listed here. He was president, as I said, of ASCB and also got the E.B. Wilson Award. He was president of the Society for Developmental Biology and got the Lifetime Achievement Award, and several other awards are listed. I was fortunate to be invited by Joe to some of these awards. On the left, you see the Roche Institute Mattia Award, where Don Brown is on the left, then there's me, then there's Joe, and then Herb Weisbach, who headed up the Roche. And what I like about that photo is Joe looks embarrassed, and that bespeaks his modesty. On the right, you see the picture of Joe receiving the Special Achievement Award in 2006, which was the Lasker Award, where Joan gave the introduction for Joe receiving that award. In addition to his science, Joe also was an amazing mentor. And at the Lasker ceremony, it was written that he was a longstanding champion of women in science. And indeed, for that, he received the AAAS Mentor Award for Lifetime Achievement, and then the ASCB Women in Cell Biology Senior Award in 2006. So the year before, in 2005, Joan Steitz presented the Porter Lecture, named after Keith Porter, whom you've already heard about from Joan. So it's somewhat ironic that she gave the Porter Lecture. And we were both sitting at the Women in Cell Biology meeting that year for their award in 2005. And Joan leaned over and said to me, we should nominate Joe for this. So that was actually Joan's idea, and it worked. And so although typically it was a woman who would receive that award. Joe is one of only 3 men who have received that award. His female trainees, among many, many accomplishments, include 4 members of the National Academy of Sciences, 3 ASCB presidents, president of the Genetic Society of America, president of the American Association of Cancer Research. Joan is co-founder of the RNA Society, and Liz Blackburn, of course, is a Nobel laureate. So let me close by sharing with you an anagram that Giuseppina Barsacchi in Pisa, who did many visits to do research with Joe, wrote. And so Giuseppina wrote, Joseph G. Gall just generously gives only science, ethics, purpose, heart, gives all, live life. A very clever anagram. And if we think about Joe's qualities for science, you can summarize them as his joy for research, excitement of discovery, keen observation, innovation, and scientific rigor. But beyond that, what made Joe so very special were his personal qualities, listed over here: generous, kind, ethical, sense of humor, modest. And so we remember Joe with admiration, appreciation, and affection, as stated in something written by Thoreau Peterson. Thank you very much.
Well, we're running a little bit late, so I will ask you to take just a 10-minute break, and then let's reconvene. At 8 minutes after the hour. Well, I'm pleased to introduce our next speaker, Mark Roth. Mark was a postdoc in Joe's lab at Carnegie. He worked on some RNPs that from Lamb-Brush chromosomes, and continued to follow up on splicing and some of those kind of things at Fred Hutchinson. But then he really made a dramatic turn and started to study hibernation and suspended animation. And this led to a lot of really fascinating findings. The founding of a couple companies, and I'm sure we'll make, Mark's talks are always extremely interesting also. So Mark.
Thank you, Alan, and Diane. Thank you for inviting me. It's a real honor to be here to speak about Joe and what a transformational thing it was for me to be in his lab. I have, and by the way, I am going to keep us on time. So one question that I have, for me, I know Joe influenced me greatly, but how did he really influence us? It is because he had a incredible passion, enthusiasm for learning. And once, as others have said, once you work with Joe, you realize you didn't really actually have a job. You were on this wonderful adventure of learning and discovery, that you were able to just embrace what nature was and what it is. And so it's with that I became a postdoc with Joe not long after he moved to the Carnegie in '83. I started in '84. And I see other names, other people in the room here who also were there. So I guess I'm representing the group of us who were there in a really euphoric time at the Carnegie. Don and Joe, Alan and Steve, among others, made it a really euphoric place to work. And so I was very privileged to be there at that time. And what I want to do is talk to you about the beginning. So I started out, I was supposed to bring ciliated protozoans to the lab, which was not so unusual for Joe. As you've heard, he worked on tetrahyme and all sorts of different things like hydra and newts and frogs and so on. And the problem was that the Carnegie at the time was not regulating the temperature so well in the summer of '84. So all the ciliates died, and I had there was nothing that I could do. My project was over. But as you can imagine, that with Joe, it's never over. And so he was thinking a lot about DNA, as you've heard, and about what the unioneme and the chromosome and the RNAs. And so when I came to the lab, I thought, we should think about what are the proteins that are present on these chromosomes, because they are certainly representing some aspect of what's going on. And so we agreed that what we do is we would hand isolate the germinal vesicles as you would to make Lamprecht chromosome sets, and then just inject them into mice, hoping the mice would recognize some of the proteins present as antigens and make antibodies against them. And then we would isolate those cells, make hybridomas that would allow us to have then monoclonal antibodies that would themselves recognize the chromosomes and particular proteins of the chromosomes, and we could think about those. So that was my project, and I see Pat in the room, others who were certainly a part of that. And here's a story of how great it was to work with Joe because of the insights that he had. And I had not realized this, Diane, that it started so young in his life, but that he was so aware that he could bring things to bear when projects became hard to pursue. So I had a monoclonal antibody that recognized the chromosomes, and it looked very striking. It cross-reacted with all sorts of different cells in plants and animals. I thought, this is just great. And it bound many bands on a Western blot, which is a way you get an indication of which proteins or what is the antigen this thing is recognizing. But there was no real way to get at what it was. So it wasn't straightforward to own the proteins that were recognized by the antibody. And so this, it was talking to Joe one day about this is really great. These are great pictures. They look nice. And I don't know how to pursue what it is that what this antibody is recognizing. And Joe recognized that when we were making Lamprecht chromosomes, those of you who have made them before, you know that what you do is you by hand, it's like peeling the nuclear envelope like you would a grape, peeling the nuclear envelope off of the nucleoplasmic ball and setting it into a little beaker, which is itself a slide, that you actually have the chromosomes sort of dancing around by Brownian motion. And they just sort of settle onto the slide, and you centrifuge the slide, and the chromosomes are basically plastered onto the glass. And then you can do things like fix them and stain them and so on. And Joe had the insight to realize that the antibody that we were using to stain the chromosomes would not stain them if we did not have magnesium present in the solution where the nucleoplasmic ball was dancing around before you centrifuged it. And without going through all of the details, it turns out, for those of you who have ever done protein chemistry, and sort of Alan showed you earlier that he saw the RNAs there in a distribution, but which ones were which and how many thousands of RNAs are there? Inside of cells, there's probably at least thousands of proteins that are there. And so this insight about the presence of magnesium as a divalent cation, the presence of magnesium tremendously affected the ability to see the amino staining with the antibody on the chromosome. So if you made the chromosomes with magnesium, they were brightly staining, and I'll show you a picture of that in a minute. And if you didn't have the magnesium, then they wouldn't. And so I went on to show, with Joe's help, how to purify these proteins from all of the proteins inside of cells by just using salt precipitation. And these proteins, which one can purify by this means, as it turns out, are the most highly phosphorylated proteins in all of cells of both plants and animals. And then, so that's how it sort of would go working in Joe's lab. You'd be stopped, and then you wouldn't be stopped because he had insights about something because he read so deeply about the subject. And it was in this way that I was able to then go on after working in his lab to show that these antigens formed a kind of discrete family of proteins inside of cells that are responsible for the regulation of alternative pre-messenger RNA processing throughout metazoans. And it sounds like a simple sentence to say now, but it was all born of what I'm trying to present is it was all born by the insight Joe had when we were struggling with antibodies that just bound chromosomes. You couldn't do anything with it. And he was so he read so deeply about things that he could have insights at moments when you were struggling that were transformational. And that was an example for me of a transformational moment. And I benefited greatly from that. And I am grateful to Joe for that. But I should say, since I'm being very positive, Joe could also be incredibly, people talk about being humble and so on, but he could also be, although subtle sometimes, he could be very critical. In fact, he could be, I like to think of it as being constructively critical. So when I came to his lab, I wrote a postdoc proposal, and he read it, and he called me to his office, and then he didn't say anything. He just got up and went to his bookcase and got this thin book out there, and he handed it to me. It was Strunk and White, The Elements of Style. And I'm like, oh my God. He didn't say a word. And that was for me a memorable interaction with Joe, another one of those kind of interactions. So there was a thing that happened in the fall at the Carnegie. I'm sure Alan and Steve and others know this. In the fall, you get the letterhead out, you're all applying for jobs. About a third of the postdocs be applying for jobs. And so I was doing that. And one of the things you would do is if you'd hope you get an interview, you didn't go give your practice talk internally, and then you'd head out on the road and hope you get a job. And so, I got mine together, and I was about to go out on the road, and I gave my talk in front of Joe, and I expected to get some deep insights, how I might, do this instead of that or whatever. And he said to me, he goes, well, Mark, there are two kinds of people in science. Those who make the most out of the least, and those that make the least out of the most. And I just walked out. I'm like, oh my God. And I think that he was always very constructively critical, but he could be very subtle with his comments. The other thing that he felt that the photographs that you had to support the evidence that you used, like that thing about the DNase-1 hypersensitives, the unanime experiment, that it had to be the right photograph, and so at one point we were trying to get a paper together, and he was having me repeat this thing over and over again, and I'm like, why? The picture is fine. No, it wasn't fine. I mean, literally, not just 10 or 20 times. I mean, each one of these Lamp-Rush chromosomes says you stain it, you take pictures. No, that's not good enough, not good enough. And I realized that what he was doing in that, although I was kind of I was a postdoc and I was pissed off because I didn't want to have to do this anymore. I thought we'd just send the paper in. But that he was trying to teach me that it really makes a difference if you do things well. Because if you do things well, then they can be remembered. And I think that's a big part of Joe's life. That when you do things well, people remember them. And that's why we'll remember him. Thanks a lot.
Make sure I've got the right order here. Our next speaker will be Zara Nizami, who was a graduate student and a postdoc with Joe. And she became a super resolution expert in Joe's lab. And really got more out of the STED than people thought was possible. And then she has continued in that vein in her career and in several companies that are now focusing in that area. So Sarah, please tell us about your experience.
Great. Okay. Well, I'm glad Mark was really short because mine's not really short. I first met Joe. Oh, wait, I got to get this clicker thing. There we go. I first met Joe when he interviewed me for grad school in 2005. I was 24 and he was 77. And it mattered not one jot that there were 53 years between us and that I had grown up on the other side of the world. We just thoroughly enjoyed each other's company from the very first moment. I can well imagine that many of us have had vivid memories of our first encounter or series of encounters with Joe, because he was an utterly enchanting individual, exuding warmth and liveliness, brilliance and attentiveness. There always seemed to me to be a gleam in his eye when he spoke, and he would wink when something was particularly bemusing. That day in his office in the old Carnegie building, we talked for an hour, which was a good half hour over the allocated time slot. He showed me his research and these two striking images just to sort of highlight that earlier point. He took perfect images and they were really memorable. And so this is, obviously this is one of nuclear bodies and then the other one loops on the Lamprey chromosome. And they immediately captivated me, as I'd never heard of them before, nor contemplated that biology could be so majestically beautiful and mystifying at the same time. So I had entered that hour thinking that I would likely attend the JHU med school program in immunology, following on from my undergrad thesis and continuing from my work in vaccine manufacturing at Merck. But I left Joe's office a budding cell biologist. I then, shall we say, stretched that first meeting just a teeny touch further from 1 hour to 12 years. Of course, I was not unaware of the general advice against staying put for too long in one place for one's training, especially considering that it was part of Joe's particularly generous brand of mentorship to swiftly train his students so that they could begin their independent careers without delay, as he did. My extended time in the Gall lab was not by any means for want of choice, as of course being trained by someone as versatile and as dynamic as Joe was like being slingshot into a world of opportunity and reaching a scientific zenith. As students, we observed closely as Joe tackled a multitude of fundamental problems of outstanding import, which he approached via the study of dozens of species, almost always whilst being an early adopter of an advanced technology, whether that was electron microscopy, as we've heard today, or Sanger sequencing, or super resolution microscopy, or to extend on what Susan was saying in these sort of eras of moving and pivoting in genomics. He learned genomics, self-taught, or indeed being the inventor of a new technology as significant as in situ hybridization. So as a result of Joe's stellar scientific training, I had a seriously tempting offer of a postdoc at the Stowers with Peter Baumann to study the Lamprey chromosomes of the parthenogenetic whiptail lizard, a dream project in a dream lab at a dream institute. But sometimes you have to follow what's in your heart and hope for the best. And what was in my heart was this immense, irrepressible daily joy that I got from the exciting science that was going on in the Gall lab, amongst like-minded colleagues, spearheaded by our fearless and well-loved mentor, Joe. To understand what I mean by the exciting science, I would like to draw your attention to this article that's coming out soon in the journal RNA. In this article, we have focused on the later Gall lab years of studying and discovering new nuclear and cytoplasmic bodies, small RNA processing, and the incredible discovery and subsequent study of cis-RNAs. It is a story full of surprising results, eureka moments, sweet anecdotes, and so many take-home messages, of which I would like to highlight two particular take-homes. Firstly, Joe was incredibly prolific during these years, never once resting on his laurels. Gall and Sveta made the salient observation that, in fact, Joe published 20 papers in the last 5 years of his career, including 3 that were prepared and published after his retirement. Secondly, this era likely contains research that is ahead of its time, with some of the earlier work on membraneless organelles already showing signs of escalating significance. I don't have a crystal ball, but I think a literature dive into the later Gall lab papers could probably tell you what to look out for as the next big thing. Having said that, Joe was never one to ascribe significance or importance to his work. He bristled from hyperbole, and his write-ups were, as we heard a little bit earlier, and his write-ups were always a simple account of the historical precedent, the hypothesis, and then the data leading to the most parsimonious conclusions. His writing style was elegant and informative, but again, there was no embellishment. Which brings me to a very important point about Joe and his scientific legacy, which was that amongst all that prolific output, he never once had to retract a paper. This is a point that he would make to me time and again. Basically, anytime I request an original paper from his wonderful filing cabinet, full of all of his papers in chronological order, because he always had a paper for something that, you know, you're like, oh, you know, I was learning about centromeres the other day. And he's like, oh, yeah, by the way, I have a paper on that. I'm like, all right. So he would stroke his hand across all the papers and tell me that this was his most proudest accomplishment. And I would politely shake my head and say to Joe, how could you be worried about such a thing? A man of your integrity and diligence. I was that sycophantic, to be honest. I really was. But it's because I just genuinely thought he was awesome. And so, but I did actually understand what he meant. Joe was humble, and he retained that healthy fear that his frequent forays into the bleeding edge of knowledge could make it difficult to get things right. I mean, nobody's seen this before. No one knows what to look for. But as we know, he's close to nature, so you can understand this a bit better. And despite this concern, Joe was in the habit of getting things right. And one could easily list so many reasons why this was the inevitable outcome. As we've discussed, Joe did not exaggerate his conclusions or use overreaching analogies to influence his thinking, always instead opting for an experiment to guide him. And boy, did he experiment. He was fully hands-on, elbows deep in all of our work in a massively good way. He was a team player at the bench, supporting us with ideas and techniques whilst also maintaining his own program of research, hence giving us the freedom to roam in our work. Joe added to this a thorough understanding of the historical literature, a deep connection to the natural world, and an impressive ability to execute experiments to perfection. So yeah, Joe got things right. An example of this was how he realized that his initial microarray results of the nucleus isolated, the hand-isolated nuclei versus the cytoplasmic fraction, when he did his first microarray experiment, the transcriptome profile looked identical in the two. And he knew this can't possibly be right because where are all the nascent transcripts and the pre-processed transcripts? He understood there were giant chromosomes in here. What would they be doing? So others may have accepted settling on the output of this advanced new technology as microarray. I'm going to publish it. But Joe applied his skepticism to the question and questioned his sample preparation. And then he went on to painstakingly remove all of this like we described, this peeling of the grape. He removed them all manually by hand. All of the nuclear envelopes gone, that contamination gone, and he got the right answer. But not only that, he got more. And he discovered this new class of non-coding RNA, cis-RNAs in the process. So Joe was good. He was really good at science, but he was also kind of just brilliant at everything. And you've already seen this picture now from Diane's talk, but he schooled me that day. And what you haven't heard is my side of the story, which is honestly, I'm a D1 athlete. I played field hockey. I played international field as a goalkeeper. I have reflexes. And I thought, and then he just this serve, and it's fun, and it was fast, and it was like boom, and I thought, oh my god, and how was I to know that he had honed his craft over the years? This is a picture from Diane, and I should have known, because I knew he was good at everything, and you know, and so that was school that day. So Joe was really fun and game for a good time, and I loved him on so many levels. So I'm not sure how to begin to describe him as a mentor and as a friend, as a central figure in my life. And I found this montage I made of for his 90th birthday that covers some of my happy memories on the left of my time in Joe's lab. And so I'd just like to add a few words to some of these points. First, this is a picture. Oh, I hope it's not too tiny. This is a picture of Joe in the actual process of teaching me and other members of the lab something really mind-blowing. This actual picture is showing us Bovary's original slides. And I can't believe that I was ever so close to so much history. But every moment, it was not just this moment, every moment being taught by Joe is precious to me. Joe spent hours and hours. I know we talk about how, he gave us this freedom, but he was hands-on in the best way. He gave me hours and hours of time at the bench, teaching me fundamentals himself personally and patiently. He taught everyone this way. A lesson in dissecting and immunostaining Drosophila ovaries, and that involved a history of the white gene. And he would show you every knob and every button on every microscope and how it worked, and adjust the gain. And so he taught you everything, how to put a scale bar. There was no minutia he wasn't involved in. And also, he left Yale for Carnegie to reduce his admin in his life. But who here hasn't been touched by Joe's dedication to his students, far and wide, on thesis committees, exam committees, he would teach a microscopy course to us just for fun. And to sort of comment on what Joel Rosenbaum's saying, he was surprised, like, how'd you come up with this 1 hour beautiful lecture? Well, Joe would tell me that for every hour of lecture was 10 hours of preparation. He worked really hard to deliver all this scientific nutrition to us. And he helped in the outreach program, and he's even on Bill Nye the Science Guy. So he's everywhere, and he was involved. And also shown here is how immersed he was in my life. And as Barb was alluding to, and I was just chatting with Barb and Stu and Lily earlier. I was like at every Christmas for so many years. And I'm right in there. And he invited me into his life, but he was also involved in my life. He knows I have a lot of relatives. I have a lot of, I grew up in Hong Kong. I lived in Pakistani, and I had no one here, but I have everyone here. And he's the third cousin coming by. And in some ways, here he is, he's wearing a Pakistani hat that my mom got him. And I say honorary Pakistani because he was so Pakistani. He had so many cousins, and he grew up with his uncle who was like his cousin, and in terms of like his brother, like his age and stuff. So I was like, well, you're a bit Pakistani, aren't you? So, he wholeheartedly welcomed me into his life. And so coming here to speak about Joe, not at his birthday symposium, as was our joyful norm, but at his memorial without him here, I have been wondering how we can bear it, and how we can process losing someone so loved, and how we can handle our grief. And I don't have the answers to this, but I do know that what I on my own instinctively did in the depths of my own personal grief was I just knew that I had to reach out to Joe and Diane, because I did eventually leave Joe's lab 7 years ago, but only because I fell in love with my lovely husband Tasneem, and I moved to the UK. And we have a very lively 6-year-old boy, Ibrahim. So this is Joe with his frog spawn, and then here's Ibrahim at a British pond with his British accent, and we got frog spawn that day, and my science life is deeply fulfilling and fun, but I had a life wobble, and though of course I have a lovely husband and doting parents and family and friends, when the tears unexpectedly came streaming down, I found a pond to sit by. And I wrote this email to him and to Diane. Dear Joe and Diane, I hope this email finds you both well. Perhaps you have heard this from many before, but it really is quite true for a lot of Gall Lab progeny that we think of you very often. In my case, even if I don't call or write or ever visit again. You both are just an embedded part of me. These days, I'm more Gall Lab than ever. In my previous update, I briefly mentioned that I switched jobs. That's the job from the super res microscope company to now at another biotech startup. I'm a principal scientist at a brand new startup that has jumped on the biomolecular condensate bandwagon. Well, potatoes on the semantics. The reality is that suddenly everything I learned about Cahal bodies, etc., and that's my term for like everything I learned in Joe's lab, mesmerizes everyone around me, and I feel like the cool kid on the block. I'm very pleased with my new role, and I still am. Every day I'm in lab, and I'm working with Joe every day. But life always has to be a tiny bit complicated. And I did suffer a personal setback in the last few weeks in the form of a miscarriage at 13 weeks. And thankfully, I'm past the physical elements of this quite well. And I felt quite emotionally tipped up until just now, which is about midway through my first day back at work. Even Cahal bodies can't seem to be abating the teary feeling I'm feeling about missing my son, whom I dropped off at nursery after having spent the last 2 weeks with him as part of my emotional healing process. But that's life. As you can imagine, I'm mostly quite cheerful still, and I really was mostly quite cheerful. My feelings today surprised me, as I had only felt tearfulness at the original diagnostic ultrasound a few weeks ago. I suspect I'll be fine again in about 20 minutes. And I was. In any case, in this moment, like where I'm deepest, downest down, I thought of you both, and I sincerely hope that life is good for both of you, and that you're in good health. And also I remember that, I have friends here from Carnegie and I should reach out to them. That's what the rest of that's about. And I did meet up with them and it was really nice. I saw Katie McDowell's light sheet microscope. So she's from Rishen's lab, and Eugene, who did the early work on the cis-RNAs, and he's at a biotech company there. So that's my news for now. Love, Zara. And as we've all been sharing, we know that Joe has left his indelible mark on us. So I can only think that gathering here today in this room and virtually across the world and time zones will help us soothe each other. Joe diligently, so that's all of us here from our birthday symposium. Joe diligently and lovingly infused all of us with kernels of his beautiful essence. So I think we will find that we will be able to turn to each other and find some comfort for our loss, just as I did sitting by that beautiful pond when I wrote to Joe, our dear friend and mentor. May you rest in peace, Joe. I will love you forever.
Thank you, Zara. Our next speaker, David Gary from the American Philosophical Society. David is a historian, and I think maybe not every one of you realize how important organizations like the APS are in promoting the production of accurate, interesting history by making collections from distinguished individuals like Joe Gall, which are not immediately, they make them a lot more accessible to historians who are working on various projects, and they can come in and benefit from Joe's life and his wisdom. So David, will you tell us a little bit about that process?
I'm a little taller here. So I know I'm the last formal talk today. It kind of reminds me of my days as a track runner, where I was running the 4x800 anchor leg. And I remember one of my coaches always telling me, get in on time and do it with some pizzazz. That's kind of my goal right now. So I know I'm sort of the one hanging up the time now. So we'll start here. So I am quite the interloper here. So like Alan said, I have a master's degree in library science, and I have a PhD in early American history, or I study the history of the book and the history of printing. So I am not a scientist, but I work with materials that I understand are very important for people, and I help acquire them and make them available to people who can use them in very serious ways. I work for the American Philosophical Society. I'm sure many in this room know the Philosophical Society. This is an institution founded in 1743 by Benjamin Franklin and many of his friends. The mission is very simple: to promote useful knowledge. And we do that in 4 different ways today. We do it through our elected membership. Alan's a member. Joe was a member elected in 1989. Our publishing arm, we have the oldest scholarly press in North America. We began publishing our transactions in 1771. And we have a grant-making aspect of what we do. We give out about $1.5 million a year. About $1 million of that is given to scientists and anthropologists who work in the field, often to people who are in the early parts of their career. And then there's the Library Museum, where I work as the Associate Director of Collections, where I manage both the rare book collections and the manuscript collections of the APS. You can see down here some of the stats of the library sort of will kind of overwhelm you, I think, a little bit. The library holds 14 million pages of manuscripts, which is roughly 3 miles of shelf space. We have 5 miles of shelving at the library. Joe added another 105 linear feet to that, to those holdings. Over the last several years, he's donated his papers. We have 350,000 books, many of them rare. We do collect other more common books, but Joe's books have come to the APS very recently, back in December, and they are an amazing collection, which you'll see a little bit of in a moment. but it's going to be a wonderful addition to what we already have. 250,000 images in our collection, thousands of hours of videotape, 225 terabytes of data. We have digital archivists on staff. About a terabyte of that data is Joe Gall's. And then we have a museum with about 3,300 objects in it. Oh, there we go. The APS also has, just to give you a sense of the type of people whose papers that Joe will be in conversation with. We have the papers of Benjamin Franklin, about 70% of the known Ben Franklin papers. The journals of Lewis and Clark. We have 800 Charles Darwin letters, the largest collection outside of Cambridge, where most of his material is. We have the papers of 7 Nobel laureates. We are promised an 8th, and we are working on a 9th. And then we've collected lots of we've collected materials from recent scientists as well. The molecular evolutionist Masatoshi Nei, Beatrice Mintz, embryologist Beatrice Mintz, and her mentor Emil Vici. Also, the papers of Clay Armstrong and his wife Clara Franzini Armstrong came here. And I can extend the Keith Porter references. Clara gave us one of Keith Porter's microtomes when she donated her papers to us. We also recently received the papers of Freeman Dyson and the last part of Dick Lewontin's papers as well. So a very distinguished group of people, and we're really glad Joe can be part of that. I want to just talk about when I first met Joe. I'd started at the APS in 2016. I was looking at many books. I looked at our copy of Robert Hooke's Micrographia, realized it was given to us by Joe in 2011. It's marked in our book. I went downstairs and talked to some of my colleagues, and they're like, "Oh yeah, Joe Gall. He has a great collection. You should write to him, Dave." And so I just did. I had nothing to lose. So I was like, I wrote a note to Joe and said, "I hear you have a wonderful book collection. I love books here at the APS. Would you mind if I come down and talk with you and visit and see your books?" It was very funny. He wrote back to me very quickly, and he said, "Dear David, no one ever wants to talk about my books with me. You are most welcome to come whenever you are free." And a couple of weeks later, I went down to Baltimore, and we had a wonderful conversation. I spent an entire day with Joe and Diane. It was really, really great. I worked at Sterling Memorial Library at Yale before I worked at the American Philosophical Society, and with Joe's deep connections to Yale, we instantly connected. Joe told me stories about how he went around Sterling Memorial Library and explored it. I love books so much and being immersed in 14 million, but we just had a great conversation about how great Yale can be and the resources that it had. During that day, he tried to teach me optics. He did a great job, I have no doubt. He pulled out lenses. He was showing me. I listened very carefully. I understood some of it, but it was very, you could tell right away, Joe wanted to be a teacher, and he wanted to teach me right away. And of course, every time I went to Diane and Joe's house, we had a great lunch too. So it was a really wonderful experience. Yeah, exactly. Eddie's, indeed. All right. So Joe gave us both books and manuscripts at the APS. Joe gave us, we talked about, there are roughly like 500 rare books, but Joe also gave us many books that were, I'd say, a little more common or just not as, or a little more popular, not the rare kind. So we have more than just the 500 rare materials, the rare titles that were brought in. The oldest imprint is from 1607, Edward Topsell's The History of Four-Footed Beasts, published in London. You'll see a picture from that a little bit later. They're mostly in Latin, French, German, and English. This is a very European-heavy collection. They were sourced mostly from I counted 27 dealers, shops, auction houses, but he also bought materials from sales of the library at Carnegie and at Yale. And it covers his interests, and it covers both his personal interests and his professional interests. Natural history, of course, microscopy, optics, cell development, cell and developmental biology. It's a really rich, deep collection. What's the next one, man? I'd just like to mention two people that he worked with. He worked with a really famous dealer named Emil Offenbacher. Offenbacher was a German Jewish bookseller who had to leave Europe during the Nazi takeover of France. He was in France for a little while, and he had deep connection to Europe and helped Joe get a lot of his really great books, especially in the early days when he was getting some of his best stuff. And then there was another really wonderful dealer named Ray Giordano, who focused on microscopy and actually was a microscope collector himself and got Joe a lot of really wonderful books about microscopes. On here. So Joe's books, these are a picture of the books that as they now stand at the APS. Takes up about 75 linear feet. We just unboxed them. They are going to be processed. That will take some time, of course. But Joe's collecting, of course, didn't start here with these really amazing books. He was a stamp collector at the beginning of his childhood. He collected insects, and he collected temporary books, like some people have mentioned along the way here today. The first big ones, of course, were the E.B. Wilson book, The Cell and Development and Heredity, Lester Sharp's An Introduction to Cytology, and Walter Fleming's Cell Substance, Nucleus, and Cell Division, which was previously owned by Theodore Boveri. That was probably his first really sort of a unique or interesting or rare book, which I know sparked an interest in him in this. But he still wasn't there yet. He was not able to, he still didn't wrap his head around the ability to own some of these kind of old materials. The next step for him was when he went to the University of Minnesota, where the head of the zoology department, Dwight Minnick, was a collector of prints and would have people over to his house, and Joe would go and look at the prints, and he realized, wow, I could actually do this too. But then Joe actually started to realize, wait a minute, all those books were broken. They were actually torn apart. That's where the prints came from. But I can buy the books and actually get more prints, and it's a much more efficient way to do things. I thought that was a very Joe Gall sort of way of putting it. He started collecting in 1966 in a small way with some interesting rare books, but it was really 1967 when he bought 40 books that were really stellar. Some of these books that would be extremely expensive today, he was able to get for very good prices. Joe sort of was in the last wave of a moment where science books were semi-cheap. So just to think about it this way, the Napoleonic era and the Napoleonic Wars caused so much havoc that all the books that sort of got spun up out of that havoc were still being sold up to World War II. And then World War II happens and books are thrusted up again into the world and moved around seriously. Many of those books started coming to America. There was a lot of money here. There was money for public universities, and there were public universities founding rare book libraries. It was a very serious moment of time where European books are coming to the States in droves. So he caught the tail end of that moment where there was so much stuff coming and where it was kind of easy to get it, that the prices were low. So Joe bought really wonderful things in the late 60s and early 70s that, even when APS couldn't afford to buy at auction today. So it's an amazing collection. He had wonderful luck with his timing as well, but he still bought wonderful things post 70s forward. I'm done here. So I'll just show you a couple of high points from his collection. Of course, there's Robert Hooke's Micrographia on the left, and Borel's book, translated, is The True Inventor of the Telescope. So of course, with Borel, this is the sort of first history of who would have invented the telescope. And what's amazing about this book, in the back of it, Huygens' discovery of the rings of Saturn is referenced in the back. It was his first way to try to get priority on the discovery, publishing it in this book. And then Hooke, of course, this is probably the most influential book of microscopy. It's the first book based entirely on microscopic observation, and the first to pair it with such profuse and interesting and just amazing illustrations. These are 18-inch long. This is a gnat. 18 inches long. These are kind of like, you open it up and it still has a sort of frightening aspect to it today. It's an amazing book. We'll come through here. So Joe donated this one a little bit earlier too. This is the pirated edition of the second edition of Newton's Principia, published in Amsterdam. It's a very important book. The first edition, published in 1687, Newton was constantly revising it, but only in manuscript until 1713 when he published it. And when he published it, there was so much interest that it was pirated right away in the Netherlands. And we have a copy of it, but Joe's copy is so much better and so much cleaner. It's so nice to have his copy and the second copy there. And then, of course, the original Mendel letters, where he does his famous P experiments and P experiments, and then basically figures out the basics of genetics. These are very highly wanted by collectors today. It comes on the market very infrequently. Joe has a wonderful set of the most important Mendel articles. It's at a price at the time that's just amazing. So really good stuff. But my favorite book at the APS, first book I came to Joe to ask about was August Johann Rosel's book on the natural history of frogs and toads, of course, with Joe's work on those animals. He collected this book. We're a little grainy here, but you can see here, we had a copy of it. It's my favorite book at APS. I wanted to see Joe's copy. Joe's copy is much better. It's hard to see in these little compressed photographs here, but Joe's copy is every book that's hand colored like this is slightly different. The colors in Joe's copy are so much richer, and just you can even just see a little bit with some of the greens on the landscape there. So Joe's copy is going to be just an excellent addition to that. And people who study these books really like to have that comparison. So I was really pleased that he had a copy, and we talked about that book for about an hour. It was a lot of fun. And of course, Rosel is just an astounding artist, an astounding scientist in and of himself. An Austrian aristocrat who was taught to be a miniaturist painter, became a naturalist when he was ill and needed something to do while he was getting better. And he taught himself how to draw and paint insects, frogs, toads. An amazing man. But the insect book was the second book I wanted Joe to show me as well. And it is just great. So this is translated as insect entertainment. So what Rosel was doing, he would publish a plate every other month, and then he would send it to his subscribers. So bi-monthly, they were getting their insect entertainment. But Rosel is known for scientific accuracy as well as aesthetic beauty. So he combines both of these in really good measure. And he wrote very detailed and very long and interesting descriptions of the bugs as well. So it's not surprising that Joe wanted to have this book. And of course, there's not just rare books in this collection. The sort of popular books, the sort of practical books, the technical books. There are a lot of catalogs that he collected from Zeist and Bosch and Lohm. I pulled out a couple here just to give you a sense. These sort of daily use materials are often hard to come by. There's ephemeral material. I'm really happy to have this material. It's just great. You can find out things like prices. You can see how people were manufacturing scientific instruments at the time. So Joe was very interested not just in these beautiful high point books, but also the sort of day-to-day science and what you needed to do to understand, to make your daily scientific work go forward. So manuscripts, I mentioned the 105 linear feet here. He donated that over the last 3 years. It includes correspondence, files on grants, symposium, travel, lab notebooks, many lab notebooks, research photographs, manuscripts, childhood material like Diane showed. I'll show you one thing quickly as well. His family material has also come to the APS. So his parents and his brothers' materials related to them are at the APS. And then he gave us an amazing letter, Raphael Maggiati to Orazio Russelli, dated 1651. It has a drawing of an early compound microscope, an improved compound microscope that was invented in 1648. Joe wrote all sorts of letters to the dealers saying just how important this is. And you can see the drawing there, one of the earliest pictures of a compound or of a compound microscope. You could just see how much Joe cared about the history of these scientific instruments. And then just to give you a sense of how big this is about half of Joe's papers here. The other half are in a different building for a number of reasons. I opened up a couple of boxes just to give you a sense. These are some of Joe's lab notebooks. There are many, many boxes of those. And then we've talked about this microscope several times today. Joe corresponded with his uncle Wilbur on his mother's side for about 5 months to try to figure out the technical side of what he wanted to have done with the microscope that was being converted for him. So these letters are full of drawings and they're full of measurements. They're just amazing. This was one of the last things he gave us. He wanted to hold on to this to the very end, but he did give it to us eventually. So that's the picture from his actual dissertation that we have at the APS as well. And then just to give you a sense of some of the things, there's Herbert McGregor's file. I remember when I came and went through those file cabinets with him, he stopped me and he said, "You're going to really like this one, kid." And he's right. It's just, I mean, I'm telling you, it's like this fat, just full of material. Joe's correspondence files, of course, are just very, very rich and very big. That is what people who come to the APS and who study the history of science want, are those correspondence files, those diaries, people talking about science with themselves. The sort of like behind the scenes conversations that come out in letters are just amazing. And that's what people want. Joe's material, or Joe's collection, is rich in that type of material. Whoops. Lastly, here we have just two examples of his childhood work. From amoeba to man, this is like a 10-page evolutionary history of man. It's a pretty amazing little book. Diane could probably give us the exact date on this one, but I mean, he must have been like second, third grade. It's very, very great. And then he kept a series of sketchbooks as well, where he was drawing different, very, very different animals. And in conclusion, so I'd really just like to end with Topsol's book on four-footed animals. So this is the elephant. Topsol's book is really amazing. It's one of these large folios. It's massive, and it's mostly a copy of Conrad Gesser's book on natural history. But that doesn't. I mean, in the end, it was a bestseller of the time. It was the top picture book in England. It's the first illustrated natural history book in English. So it's not surprising that Joe wanted this book. But I pulled out the elephant for a very specific reason. So famously, elephants don't forget, but people do. I have the best job stewarding collections like Joe's. So archives and books are tools of remembrance, I'd argue. So Joe, we talked about Joe being on many of you. These are things I wrote. I met Joe, worked with Joe probably about a dozen times over the last several years. I could right away tell he was dedicated, collaborative, a generous mentor, patient. These are all words that I came up with before I came here today. We're all bringing them up over and over again. I think Joe's archive will help us to remember these values, especially as we navigate the massive challenges we're experiencing today. Joe wanted to do things right. I think about that all the time. Work hard. My parents taught me that. How hard you make sure you follow all the right steps. Share and be collaborative and be positive. Joe encompassed all of that. And the books. I'll finish on that. So how you can interpret book collections in many different ways. So I'll end on this one, my interpretation. Joe was looking back to see where he could fit in and where he could contribute with his collection. And what I'd like to think is that all those authors would say to him that he contributed and that he deserves a place with them. So thank you all very much.
In light of the fact that we're a bit behind schedule, we were thinking that we won't have time for the open mic, and we'll need to go to our final speaker, Steve McKnight, who I hope is here. Oh yeah, there he is. So Steve came into Joe's orbit very early because he did his graduate work with Oscar Miller at Virginia, and Oscar Miller had been a postdoc with Joe, and I think there was some interaction even while Steve was there. And then he came to Carnegie either as a postdoc or a staff associate. I never could get it quite right, and there wasn't much difference in those days. But he worked with Don, and he made some fabulous advances in the area of transcription factors. Then after a brief period away, he came back in 1983 as a staff member, right about the time Joe got there. And Steve has thought a lot about what's important in science and, you know, what we need to keep in mind. And he's going to share some thoughts in conclusion for all we've heard about Joe and about how important it is to do science right. So.
This is really special for me. I mean, I could look out here and I see Mark and Joan and Alan and all these people, and these are like a family. I mean, they're the family of Joe Gall and their family of Don Brown and Diane right there. And to be a part of this family is extremely special. I'm a lucky person. So here's what I thought about. I thought about, what am I going to say? And I'm going to say the title of my talk is The Genius of Joe Gall. So in the 1950s and 1960s,
the field of molecular biology was just getting going, and it was mostly derived from work on prokaryotic organisms, bacteria and their viruses. And the superstars were Jacob and Monod and Windsor and Letterberg and the phage group at Cold Spring Harbor. And even Marshall Nierenberg used bacterial lysates to do in vitro translation to crack the genetic code. So the early time of molecular biology was almost exclusively in prokaryotic organisms. But in the 1960s, a small group of people began to try and evolve into the field of eukaryotic biology. And there are a number of people who are in this group, but the ones that I remember are Max Bernstein and Bob Perry and Jim Darnell and Don Brown and Joe Gall. So by serendipity, many of us who are here today fell into the orbits of Joe Gall and Don Brown. And for me, it was, as Alan said, having been a student of Oscar Miller. So I'm kind of a grandson of Joe Gall. And so I fell into this orbit by serendipity, just luck. Then to further my luck, I was able to be here in Baltimore at the embryology department between 1977 and 1992, the formative time of my career as a scientist. And I got to work with both Don Brown and Joe Gall. Don was the director of the department for 15 years, and Joe was there for 10 years. And these are my heroes. It was unbelievably good fortune to be with them during that time. So when I look at it, the Venn diagram relating Joe Gall to Don Brown, it overlaps incredibly. And the overlap is because they had the same values. Now, they weren't identical. They're different in distinct ways, but they had the same values. And we are the beneficiaries of learning those values from those two wonderful people. So you've heard from Joan and Alan and Mark and Joel Rosenbaum and Susan and Zara and others, especially this last talk we heard from the wonderful talk about Joe's collection of books. Today's has been fabulous. So what you see is woven together a tapestry of who Joe is. And I can't add a whole lot to that. I would say just exactly the same things because it's very clear who Joe was. So what I can do is kind of what Joan did. I can tell you a few things from my own perspective, more somewhat about me than it is about Joe, but that's what it is. I'm not going to tell you much that you don't know, but I will tell you a few things that may surprise you. So in thinking about Joe, I come up with three things. One is sensibility. He had a great sensibility, not complex, just sensible. The second thing I think about Joe is scholarship, and we all know about Joe's scholarship. And the third thing is curiosity. So in that order, I'll tell you a few of my thoughts on Joe. So let's start with sensibility. So in the 1980s, Joe was sent a manuscript to review by Ben Lewin at Cell. And the manuscript claimed that a chromosome, an extra chromosomal piece of DNA of Chlamydomonas was packed into the basal body. And Joan talked a little bit about the basal body, and Joel Rosenbaum knows all about the basal body. So Joe got this really cool manuscript that said that this huge piece of DNA, 10 megabases in size, was packed into the basal body during mitosis. And this was really cool. Like, here's this extra chromosomal piece of DNA that encoded flagellar genes, and it was all bullshit, but forget that. It was really cool to think that it could be packed into the basal body. And so he was asked to review the manuscript. And so what did Joe did? Joe went straight to micrographs of his own and looked at measured the volume of the lumen of the basal body. And the size of the lumen was such that if you stuck in the billion Daltons of this supposed chromosome into the basal body, it would be more dense than a black hole. So sensibility told him this can't happen. So he sent in his review, and 2 months later, the paper was published. On the cover of Cell was a basal body. It was clearly wrong. It was idiotically wrong. And how did Joe know it was wrong? He knew it because of sensibility. Just measured size and it wouldn't fit. And of course, it turned out to be wrong. Susan Dutcher later showed that there was no such thing as this unique chromosome and so on and so on. But that was the first example that I remember of Joe's sensibility. A second one came about on a picnic with Diane. And we went to a pond, and I can't remember the pond, but we're walking around the pond. We had a picnic and looking at the pond, I saw a clutch of amphibian eggs, a nice clutch. And I said to Joe, I said, geez, Joe, how many of those do you think will end up being fertile adults? And he said, Steve, are you stupid? I mean, he said, if this population of frogs is in equilibrium, steady state, the answer is 2, 1 female and 1 male. And so that's just sensibility. That you know, didn't take brilliance, but Joe was just sensible. And it really helped me to stop and step back now and then in science and think, does this make sense on kind of first principles? Before going forward, let's think about sensibility. So now I'll move from sensibility to scholarship. I don't think I ever knew anything that Joe didn't already know. Never. Maybe I knew something about the Baltimore Orioles that he didn't know, but I'm talking about substantive knowledge. I never knew anything that Joe didn't already know. And you know, the only way I figured that I could know anything that Joe didn't know was to discover it. Because if it was there in scholarship, he would know it. So I'd never beat him at that. Anyway, so he was an inspiration. So here's an example of the depth of Joe's knowledge. So again, it was with Diane. She and Joe came to dinner one night at our house, and he looked up on the shelf of a bookshelf and saw a book that on the side of it, on the edge of it, said, The McKnights of Snickersville. And he looked at me, he looked at the book, and he said, can I take a look at that? And I said, sure. And he took it down, and he's looking at it, and he said, geez, Steve, I had no idea your family was from Snickersville. And Snickersville had not been on the map since 1900. The name of the village had been changed from Snickersville, Virginia to Bluemont in 1900. So my father had written this history of our family, and he had gotten back to Snickersville, and the only way he could find it is by finding maps that were before 1900. So how in the world did Joe Gall know about Snickersville? And he said, oh, Steve, Snickersville is right where Snickers Gap comes out of the Blue Ridge Mountains. You know what I mean? Jesus Christ. And then he said, that's where the Civil War battle of Cool Spring happened. So he knew that. And he said, and to boot, it's a great place to bird watch for hawks and pileated woodpeckers. Joe Gall knew everything. He was unbelievable. So I'll stay on scholarship. And Zara and Joe and I wrote a paper in 2017, okay? And I've always tried to take pride and try and write papers very clearly and precisely, but I didn't know what I was in for when writing a paper with Joe Gall. Every word had to be perfect. I mean, absolutely, really well thought out. And the references had to be scholarly perfect to the right reference, appropriate. I mean, the degree to which the seriousness to which he took this, it was just at a wonderful level of scholarship. Okay, so the last thing I'll talk about is curiosity. And so I only have one example of this. And it was, I think Mark was there at this time. So I walked by Joe's office. My lab was at the end of the hall and Joe's was right here. And Joe had a microscope in his office, if I remember right. So he's at the microscope and he has a reagent kit from Bethesda Research Lab. on the bench next to his microscope, and he has his pipette man, and he's mixing parts of this blue tube with this red tube and putting it on the microscope. And this is a reagent kit, maybe GT11 cloning kit or something. And I'm saying, I say, Joe, what are you doing? What do you, I couldn't imagine what he was doing. He said, Steve, when I mix these two together, it gets cloudy. And I just wanted to know whether it was a crystalline precipitate or an amorphous precipitate, as if that mattered to anything. But that's Joe. Joe's curious. He simply wanted to know. And I say that because 25 years later, I had a chemical that I put into a lysate of mammalian cells, and that chemical precipitated all of the transcription factors and RNA binding proteins of the lysate. It was like magic. It was like voodoo. I put it in, spun it down, and it left all the other proteins and everything in the cell, but took down all of the cool proteins, Jones RNA binding proteins, my transcription factors. This chemical magically brought down everything. How in the world could a chemical precipitate hundreds of different proteins? And so I did what Joe did. I went and took it, and I put the thing on a microscope slide. I went in, and it was a crystal. It was a crystal. And so we saw the crystal at 0.9 angstroms resolution, and it had troughs in it, perfect for beta strands to fit into. So by listening to Joe and following curiosity, I could solve something. And that was really one of the coolest things I ever observed in my career. And I learned it from Joe. And that is to be curious, to have curiosity. So those are the pillars. So I'll close with an anecdote and then a summation. So the anecdote comes like this. After I finished with Oscar, I got to go over to Europe, and I visited John Gurdon's lab, and I visited Heidelberg and different places. I got to meet William Barnhard. It was one wonderful time. So in Heidelberg, I met a guy named Uli Scheer. And Uli Scheer was a really good guy. And I was there to try and help teach him Oscar's technique of chromatin spreading. And Uli Scheer was dissecting his amphibian oocyte germinal vesicles. And he's doing it in a ringer solution. And I said, I'm wondering if he might use the same ringer solution that we used. And of course, we learned that ringers from Joe Gall. And I said, what buffer are you using? He said, of course, we're using St. Joseph's water. I mean, so Joe was worshipped. He's literally worshipped. So I've long tried to grade myself with respect to what I call a CA ratio, and that is curiosity to ambition. Okay. So to me, the greatest scientists have a really high ratio, high curiosity, not worried about ambition. And I never met a person with a higher CA to ratio than Joe Gall. Never. Okay, so several years ago in this room, we described Don Brown as our North Star. He was our Polaris. And so now I offer the symmetrical thought that Joe Gall was our Sigma Ontatis. So what is that? That's the motionless star of the southern sky. So we have the northern pole up here, Polaris. So there is a Polaris of the southern sky. And I'm going to call Joe that pole. So between the two of them, Joe is a North Star and Joe is the South Star. They formed what for us is a gyroscope. The Earth's axis spins on that axis. And those of us, because of their leadership, created something that is a gyroscope for us to have stability. And they gave us something so special. So with that, I'll quit and thank you all for listening to me this afternoon and being with Joe.
Well, that's just been a terrific experience this afternoon. Thank you all for being here. I'm sorry we didn't get to hear all your voices that as we had hoped, but we do have, and we're already a little bit late, to head back to the Carnegie, and there's going to be some refreshments, and we look forward to talking to as many of you as I can there. And so thank you all again, and thanks to everyone who made a presentation, and we'll see you back at Carnegie.